become an expert in taxonomy. So, with this background only, we have given the title Ichthyotaxonomy, Foundation of Fisheries Science. So, why this is important? The report says that the number of organisms in the world may range from 10 to 100 million. It may be 10 or even up to 100 million. So no one knows correctly. But what the scientists have discovered so far is just 1.7 million, roughly 1.7 million. If the total is 10 million, we have discovered only 70 million. If it is 100 million, we have discovered only 1.7% of the total number of organisms that are existing in the world. Then it means that we need to discover still 85, more than 85 or 97%, 80% of the organisms that are in the world. We need large number of taxonomists. Actually, we don't have these many number of taxonomists. It will take several years, thousands of years. So meanwhile, if we take a long period to discover all these things, many organisms will go extinct or endangered. It won't be of any use to the ecosystem or to the society. So we have to try our level best to discover all the organisms that are available in the world, because some are important for the medical field, some are important for the fisheries sector, some are for agriculture sector, for humankind. So we have to try our level best to discover all these unexplored organisms, identify these organisms. But at the same time, we have a shortage of taxonomists. What? Because most of the people, they don't come forward to take this as a subject, because they don't get proper recognition on. So to fill this gap, to motivate them, to bring many people into this field, so international level, people started worrying about this and they started organizing this day to motivate them to appreciate the taxonomists around the world. So that's why we are also organizing. We have been organizing this World Taxonomist Appreciation for the last 4 years. So I think this is the 4th year. So now for this wonderful program, so we all have gathered here. But first and foremost, I wholeheartedly welcome our beloved Dean, respected Dean Dr. Ahilan, so who has been instrumental supporting and providing motivation, guidance, facilities to organize this sort of programs very often. So on behalf of our department, on behalf of all the taxonomists, I wholeheartedly welcome our Dean to this organization. So if you organize this sort of program without a taxonomist, so our purpose won't be served. So it won't be fulfilled. That's why we have an eminent ichthyologist, eminent taxonomist, expert in population dynamics and stock assessment. Above all, excellent teacher, mentor, and teacher for most of our senior faculty. So none other than Sir Dr. Venkataramani, our former Dean and Director. So on behalf of our department, on behalf of all the taxonomists, I wholeheartedly welcome our Venkataramani sir. Thank you, sir. So next, I would like to welcome all the heads of the department, esteemed colleagues, my dear students, the non-teaching staff members. I sincerely welcome all to this wonderful programs. Please, students, so we have 3 speakers, eminent speakers in this field. One is Sir Dr. Venkataramani. He's an expert in taxonomies. So he'll be starting with his lecture on taxonomy principles and the classification of marine organisms. That will be followed by freshwater fish taxonomy by Mr. Moulitharan, Dr. Moulitharan. So he has been working in this freshwater taxonomy for the last 6 or 7 years. He has very good practical experience. At last, we have a lecture on modern technological tools that are available for the identification of organisms. That will be delivered by Dr. Suresh from IT. So try to be present here for till the end of the program and get benefits out of this program. So at last, I thank our Dean for having given us this wonderful opportunity. So thank you.
Thank you, sir. And now I invite our beloved Dean, Dr. D. Ahilan, sir, to present the inaugural address to the class.
Our most respected Adjunct Professor, Dr. Venkataramani sir, my teacher, my guide and everything, the coordinator of this program, Dr. Jayakumar, Organizing Secretary Durairaja, my dear faculty, beloved students. Very good morning to you all. March 19th is the Taxonomist Appreciation Day, a time of the year when we can thank all taxonomists around the world for the hard work, sincere and other. Taxonomy is the branch of biology in charge of classification, naming, description of existing organisms. As our professor rightly pointed out, it is estimated that there are more than 8 million to 1 trillion species available in the world, existing in the world. 8 million to 1 trillion species are available in the world, and so far we have named only 1.6 million. So as you all know, this taxonomic process is very slow, as often only experts in the field can name the species. This can take years, as they will study the combinations of the form, shape, structure of organisms together called as morphology, with the behavior, genetic, and biochemical process. The taxonomy is the branch of science that deals with the studying of organisms, classifying of organisms. Classical taxonomy deals with the classification of species in which species are considered as a basic unit. Further, an organism is classified into domains, kingdom, phylum, class, order, family, genus, species, and species. In addition, the organisms are classified using both morphology and phylogeny. Carl Linnaeus, you might be knowing Carl Linnaeus, the father of taxonomy, was a Swedish zoologist who created a new system of classification for all living organisms. His first attempt in classifying plants and animals was published in the year 1975 as a book called Systema Naturae. His system of naming, ranking, and classifying organisms is still in use with little modification and all. So the World Taxonomist Appreciation Day, March 19th, was celebrated from the year 2013 to make a point that the sincere work of all taxonomists needs to be appreciated in the world. Taxonomic scientists follow rules that govern process of classification and identification to ensure that their work is able to be adopted anywhere in the world. That is very, very important. So if you find a species here at Thoothukudi, it should be identified in the US also. That is the classical example for taxonomy. The global literature shows that there are more than 1.1 to 1.5 million described species, with the estimates for the total number of species in the world ranging from 5 to 10 million. With all the taxonomic work to be done, it became as a surprise to learn that taxonomic themselves are endangered. It has been estimated that to discover and describe all the species, what I said, there are more than 10 million. So if you want to describe all the species, it requires more than 3 lakh taxonomists. If you have 3 lakh taxonomists, you can classify all the organisms. And also it will take more than 1,200 years. And we need a budget of $350 billion. US billion dollars. Then only it is possible for us to describe all the species that are existing in the nature. So taxonomic work is the foundation for understanding how to save that we can make plans for the future. Further, to fix the taxonomic crisis that requires recognition of the essential nature of the work of the taxonomists, the value of herbarium and museum collections that those who run them to exploit in the world. Taxonomy is critical for almost everything we do in biology, and as a community, we need to support taxonomy and its practitioners. In our fisheries sector too, there are several ichthyotaxonomists, namely Francis Day, India, Peter Atleti, Albert Gunther,
William, they played a major role in the classification and identification of aquatic organisms that paved way for the development of fishery sector and also for taking measures for sustainable exploitation and conservation of fisheries resources. As far as our college is concerned, we have got a lot of taxonomists like Dr. V.K. Venkataramani sir, the person who is available here, then Venkataramani sir, Dr. Ramanan. So they are all the very specialists in taxonomy. And I request our faculty to kindly make use of their expertise because our sir is available here itself. So whenever you go to taxonomy class, you can take it for 1 day for practical. So his expertise needs to be available. Okay. Because others to the Ramanan, the V.K. sir is available along this distance. Because he is available here, we have to make use of his expertise. So we have been celebrating this World Taxonomic Appreciation Day on March 19th for the last 3 years and by arranging special lecture. In this year, we actually planned to celebrate the World Taxonomist Appreciation Day by organizing a national webinar on Ichthyotaxonomy, the foundation of fisheries science, with the special lectures experts in the field like Dr. V.K. Venkataramani, Dean retired, who is going to deliver lecture on classical taxonomical methods and taxonomy of marine fishes, and Dr. E. Suresh, assistant professor. I'm sorry, I didn't mention his name. Dr. E. Suresh, assistant professor and head in charge Department of Fish Genetics and Breeding, IFPGS, TNJFU, Chennai, will be delivering a lecture on application of molecular tools in taxonomy. I welcome on behalf of our college, and I also welcome Dr. N. Moulitharan, assistant professor contractual, Dr. MJF TNJFU, Pondeli. He will be delivering a lecture on taxonomy of freshwater fishes. When I was at Pondeli as a dean, I witnessed his work because he will be always in the field, in the Arani River and several rivers. So his work needs to be appreciated, and he got PhD also in this freshwater taxonomy fishes. So I really appreciate the organizing team for organizing this program in a nice manner, and I request all the participants to make use of this platform to enrich their knowledge in the application of taxonomical tools. I once again thank our Professor Dr. V.K. Venkataramani for his work and all. I thank one and all for being present here. Thank you.
Thank you, sir. And now I invite Dr. V.K. Venkataramani sir, Adjunct Professor and former Dean of our college, to felicitate the gathering.
My good morning to you all. Whether it is a taxonomy appreciation day or not, but my beloved student, our Dean, Dr. Ahilan, and my beloved student, Dr. Jayakumar, has given a more appreciation to me. But whether I deserve it or not, it is to be decided by the Almighty. So, I am happy to be here. So, at the foremost, I tell something about the importance of the taxonomy. Before that, let me go for the formal inviting my beloved students and also some of the eminent teachers like Shalini, Rani, and Mama Geshwari. They are all sitting here and they are all my beloved students. Understood. And also, I am happy to say that this is my 45th year of my continuous teaching. Right from 1981, I was appointed in Agricultural College in Madurai. Then I was transferred to Inaugural in 81. And even after my retirement, it continues. Of course, Almighty will decide me. So, because I am having a small amount of experience, I am not that much capable or competent to discuss about the taxonomy because it is a vast subject. And in this important day, we have to remember two eminent personalities. One is the father of Ichthyotaxonomy, who died at the age of 35 himself in a car accident. And we have to remember the most famous man who has introduced the binomial system of nomenclature and the These two people are to be remembered and remembered forever, as long as the taxonomy is there. Because Father Atreides was a church monk, so to say, and when he was sitting even in the church, he has to write many number of books about the invention of fishes, particularly in the Atlantic and also in the Indian Ocean. Of course, he died in a car accident at the age of 35, a premature death. And with regard to Linnaeus, he has made a tremendous breakthrough with regard to binomial system of nomenclature. Now, if you take the taxonomy today, in our days, we study only on the morphometry, morphology, etc., osteology. Now, the taxonomy has moved to different angles, because the invention of many softwares. Understood. Now, the current study is the autolytic chemistry. Understood. Our own conventional morphology, of course, it's all important, no doubt about it, go for the beta identification because morphology is a must. Understood. But now it has put on to what you call it as at the lab-oriented studies, particularly the genetics and the molecular studies. So the taxonomy has become, nowadays it has become an integrated approach, so to say. So based upon this integrated approach, you go not only for detecting the species, but also you go for the evolution of the species, and also you go for the phylogenetic classification of the species of the family, so to say. And many of the orders, many of the families in our times, now it has been totally changed. Day by day it is changed. But remember, all the old classifications are not outdated, they are the ridiculous. Understood? From which the modern taxonomists can travel. As far as the taxonomy is concerned, it is only professional. It is liable to change daily. It is not a military rule, so to say. It is not a law. It should be like that. It should be changing because many taxonomists are coming with a new invention, with a new molecular characterization. So, taxonomy is a subject which is so dynamic and it is having its own heuristic values, exploratory values, and also predictive values. And apart from that, taxonomy is a professional one. It is always liable to change. Understood? Every time if you see new classification, when we were studying, we studied the classification of Leo as a bird. Then it has moved on to Weber and Dewey. Then it has moved on to Francis Day. Then now the current classification is a completely different classification go for the molecular aspects. So I advise the young students to concentrate both on the classical and also the experimental taxonomy, so to say. Because of this, you can go for the nice phyletic erection of a new taxa and also new family at the higher taxon level. So this is a fascinating subject, so to say, and my own head of the department pointed out that taxonomy is becoming outdated. It is not becoming outdated. Day by day, when the biotechnology is coming up, because I was fascinated to study the autolytic chemistry. Previously, we used to take the autolytic for identification, that's all. Inner view, outer view, circles, all those things. Now, the entire chemical composition of the autolytic, they go for the identification of the new stock itself, even at the lower taxon itself. So, day by day, it becomes more fascinating, and also after the invention of these so many softwares, the best morphologic analysis is also gaining much importance. So this is a fascinating subject, and anybody who is interested to study the taxonomy, I advise you to go for an integrated approach than what we did in our times, studying only the classical taxonomy, and this will not be that much useful. So we have to go for the integrated taxonomical approach, and that will be a very meaningful one. So with these few words, let us remember Atreides and also Linnaeus and also some other eminent taxonomists. I will not say the taxonomists, they are the voyage scientists like Cuvier, Valencianus, Forrestal, then Gilchrist, Thompson, because these are the people, they went for a very good voyage, Whenever you just see any taxonomical books, Revere and Valencies, Forrester, Valencianas, then Gilchrist, Thompson, all these people will be there. So those eminent voyage scientists ought to be remembered today. So with these few words, let me conclude my felicitation first. I once again thank the authorities of the TNDAQ and Professor Ahilan, my own student and beloved Dean and Dr. Jayakumar and faculties of this department for giving me a small opportunity to talk on this subject. Of course, I was here in this Taxonomist Appreciation Day, if I am correct, right from 2016 itself. I am giving the lecture till today for Taxonomist Appreciation Day. I am here. Understood. First, David Kingston called on me, then Dr. Venkataramani called on me in Kanyakumari, and then Paul called on me in Thoothukudi. Understood. So, thank you very much for your what to say for your presence, and also I thank our staff for giving me an opportunity to tell something about the importance of classical taxonomy and also the identification, some of the commercially important verifications. Thank you. Thank you, sir. And now, I invite our beloved Dean, Dr. Ahilan, sir, to present the memento to Dr. Venkataramani, sir, Adjunct Professor, as a token of gratitude. And now I invite our professor to present the memento to our beloved Dean. Thank you, sir. So now I invite Dr. R. Durairaja, sir, Assistant Professor and Moulitharan Secretary for the National Webinar, to present the vote of thanks.
Very good morning to one and all. And we are at the end of this inaugural session. First of all, I want to thank for this opportunity to propose a vote of thanks. So on behalf of DFDRM, I wish to extend my heartfelt gratitude to the most respected head of the institution, Dr. B. Ahilan, sir, for his continuous inspiration to conduct such a wonderful program like World Taxonomist Appreciation Day 2024. So in order to honor taxonomists, so this time also he has made it very simple, and he had given the permission to organize such a program here. And he also pointed out the importance of taxonomy and taxonomists, how to identify, classify, and naming the fish species, very important. And next, I wanted to extend my heartfelt gratitude to Dr. V.K. Venkataramani, sir, former dean and adjunct professor of our department, who has been with us more than 4 decades. Is it? So he is a well-known person for all of us, as he taught taxonomy and other related courses during our study periods. And he has been readily accepted our invitation to be with us for this wonderful program. So he insisted in his felicitation address that various taxonomical methods and its history, and he emphasized on conservation and management of fishery resources. I would like to thank our beloved speakers for this event who has been invited. That is Dr. E. Suresh, Assistant Professor, Department of Fish Genetics and Breeding, and Dr. N. Moulitharan, Assistant Professor, Contractual, Dr. N.G. Ponneri, for accepting our invitation to deliver a series of lectures with a special emphasis on fish taxonomy and its importance in the modern era. So I wanted to thank our HOD, who is being motivated and determinant and perseverance in coordinating various activities to conduct a nice program like this to be successful in a grand manner. So further, I would like to thank Dr. C. Sudhan to be act as a MC for conducting this event in a very grand manner. And I really wanted to thank Professor, Associate Professor, Assistant Professors of the various departments of this college, and who spared valuable time to create this occasion. It's really unforgettable one. So in addition, so we would like to thank our students. So without them, it is unimaginable. So then I thank Mrs. Prema, Ashok and Yasaki and Anto and the rest of all our department staff. At last, I would like to thank Dr. N.V. Sundakumar sir, Professor and Head Dean, for permitting us to use the AV hall facility. Further, I thank Jayaraman Ramakrishnan, Deputy Warden and JE for his support to organize this event in a good manner. Last but not least, on behalf of DFBRM, I wish to thank all alumni team for being connected to the program without any kind of disturbance. So I thank once again.
Thank you, sir. And now the three lectures we are having. So the first lecture will be delivered in hybrid mode by Dr. D.K. Venkataramani, sir, Dean retired and Adjunct Professor of Department of Fisheries Biology and Resource Management, Fisheries College and Research Institute, Thoothukudi. The topic which sir is going to deliver is classical taxonomical methods and taxonomy of man. The lecture will be delivered in hybrid mode.
Good morning to you all. And, so this is the topic which I am going to take. It is a very elaborative topic. It could not be taken for even one semester. So, since a lot of time is 1.5 hours, something like that, understood. I will be giving the importance, of course, some of the topics in this lecture might have been already dealt by your teachers, even at the undergraduate level. So, the topic is the classical taxonomical methods and also the taxonomy of narrations. So, if you take the need for the taxonomical studies, as I told you, it needs mostly an integrated approach. So, the principal task in fish taxonomy is to go for the identification, classifications, and study of species formation. So, one has to go for identification and classification. Identification, you can do it by a deductive procedure, and classification is by inductive methods. And based on the deductive procedures, one can adopt for the classification of not only aquatic taxa, but for the case. Any taxa, even at the terrestrial side. Now, what is the most prerequisite and the importance of both in the molecular approach and also in the classical taxonomical approach, particularly in the identification of the species, where if it is a finfish or if it is a shellfish, whatever it is, then you have to concentrate on all the length groups and also their sexes. And this is to be collected in adequate number of samples, because if I am able to see most of the new species description and also new redescription understood by many of the taxonomists, particularly from Indian side, the sample number is very less, and also they go for the erection of a new species and also with less number of samples and without going for any sex coverage or the length group coverage. with the 2 or 3 specimens the species are erected, and that should not be the case. Understood? And locality collection are more meaningful than restricted sampling. And remember, whenever you go for the redescription of species or the new species erection, whatever it is, you have to follow the set of principles formulated by the International Code of Zoological Nomenclature and Seasoning. That is, we call it as code, CODE, understood. That has to be properly, it should be followed. We will go for the law of priority or emendation or with the signaling or with what you call it as with the typological concept, holotypes or the parallel types. So the paramount importance should be studied, particularly the holotype of the specimen should be studied. If you want to go for any description or any classification or erection of any species, you know, where the holotype of the particular species, which is deposited in a particular museum, should be studied carefully. Then only you can make a clear-cut differentiation. Nowadays, none of the taxonomists, you know, whenever they want to redescribe a species, they never concentrate on the holotype. They just see on the identification books and they come to the conclusion, and that is not the case. And the holotype, or even some of the paratypes that has been deposited, it should be strictly followed. Then the character evaluation should be big. So if you take the classical taxonomical methods, you have got the 6 type of methods. We call it as the classical taxonomical methods. One is the external morphology, the other is the conventional morphometric, the third is in the last 2 or 3 decades, we have the morphometric studies and the heuristics, the osteology, and also the soft anatomical studies. We call it as the classical taxonomical studies. Remember, classical taxonomy studies has got its own merits and demerits. I cannot call demerit. It has got its own limitation. But at the same time, the molecular taxonomy is not that much advanced as most of the biotechnological. It has got also its own demerits, so to say, because I have employed many people in National Bureau of Genetic Resources. Understood. Even in electrophoretic studies, particularly the muscle agents or iron protein or mitochondrial DNA, whatever it is, you know, well, the sampling is there. One such minus point, it has to be more much sensitivity. Understood. And also the collection of specimen, everything should be there, because I myself have seen when I was running the electrophoretic studies in islands for me, locality wise there was a different electropyrograms, understood, particularly both in the islands protein and the muscle myelin. So that could not be also be taken as a valid one, but an integrated approach can be made even to showcase it with the amino acid profile also. So both the classical taxonomical and the molecular taxonomical studies has got its own merits and demerits, but what are all the demerits that has to be analyzed most? For example, the chemical preparation is the most point, particularly in the biochemical taxonomy and also in the molecular taxonomy. And for our studies, you know, the classical taxonomy, just taking 2 or 3 specimens and coming to a very good conclusion that it is a new species because the dorsal fin is 28 spines or 30 rays, something like that, and going for a redescription and going to some of the journals like the Nasarated journals in Pakistan journal and getting 6% and getting published. Of course, for your career purpose, it is very good because you will be elevated also or professor and you may get the appointment. That is not the case. Understood? Because why I am saying is when another redisclaimer comes, you know, you will see the whole lot. Understood? And you will put the new species selected by you in the dustbin. Understood? You will be getting the name, but it will be in the dustbin. So it has to be very critically followed when you are using the classical taxonomical approach. The first study you take it is the external morphology, and many characters are there. And when I am telling the some of the important characteristic features of the marine families, because as you all know, if you take the marine fishes, if you take in Indian waters, there are more than 2,500 species are there. So, and we have got about more than 63 orders, and of which the order Perciform, now it has been divided into many suborders and even orders. Understood? But the father of the order, the Perciform, which includes 55 families, now the Perciform order has been divided into many suborders. Understood? So I will be dealing mainly on the major families of the order Perciformis, Dean and Perciformis. I have to put that word, and also the Cruciformis, and also the Pleuronectiformis, and also what you call it as the Siluriformis. I understood. So because these are all which I feel all are commercially important, which I feel these are all the commercially important groups, and that has to be finished in the stipulated time. So if you take the external morphology, well, these are all some of the characters, that could be of very good use, particularly in the identification of taxa. Remember, when you go for the external morphology or for the morphometrics or for the meristics, well, in the linear classification, we have got higher taxon and lower taxon level. So in the higher taxon level, up to genus taxon, well, this external morphology could be useful. But if you take the lower taxon, either at the species level or at the subspecies level or even at the stock or the race level, well, the molecular taxonomy will be of that much useful. And also, so is the case with the meristics and not only chemistry, osteology, and also the trust morphometry. Now, coming to these external morphologies, that could be useful both in the lower taxon and also higher taxon. These are all the characters understood that could be of very good use, particularly in identifying a genus or a species, which I will deal with when I am doing the family characters in the next. Then there are also some merits and demerits, particularly when you use the external morphology. Why I am telling is the color variation is more abundant because there are two species. Just this I want to tell. The usage of external morphology, the Caranx stellatus and also Caranx melambicus. Understood. The Caranx melambicus and stellatus, well, the pigments will be present up to certain range, and this will fade off. Understood. And it will become small marks. So previously, these the same species it was identified with the two species. One is stellatus and the other is melambicus. Understood. Now, because of the colorations, it also poses good amount of problem. Understood. So it has to be studied critically. Then later on, seeing the entire length group, understood, of this stellatus species, then the melambicus was made as a synonym to that of the stellatus, and it was treated as a single species. So whenever the taxonomist wants to give any importance to coloration, of course, coloration is good in some angle because the coloration, particularly for the identification of some of the reef fishes, particularly the Catodons, the Pogocentrics, and also the Acanturids, well, the coloration goes important point. At the same time, the coloration also changes. For example, if you see in the case of the Pogocentrus semicirculatus, which you popularly call as the Allah fish, understood, where in the caudal fin, the Arabic numeral markings, they are liable to change once a fish reaches 15 centimeter and above. Understood. So this is only the pigment changes. Understood. This is in the form of the Arabic numeral. So the species which is less than 10 centimeter in the light conditions, it may be fetching more than 2,000 or 2,500 rupees. When the pigment goes up, it comes to about 600 or 500 rupees. So these are all some of the drawbacks of the coloration. But at the same time, the coloration, particularly if you identify some of the important families like Phalidae, Phalidae, and also the family Libectridae, where the coloration is important, particularly in the reef fishes, according to the habitat, the coloration changes. For example, if you say the seahorses, with regard to the seagrass beds or the algal beds, the coloration of the hippocampus will change. And if it is in the coral beds, it shows an excellent amount of camouflage. So these are all some of the points that are taxonomists we have to remove. Next slide. Then coming to the morphometrics, we have the two types of morphometrics. Morphometrics, of course, you will teach right now. It is widely used in fish taxonomy, which I may not go in detail. And you have to the measuring board also the teachers might have told you, and the fish should lie on the right side and the snout should be on the left side. Understood. Either in the wooden scale, the measurement should be recorded at point 5 mm with a fine craftsman divide. Understood. So the measurements, too much of measurements it will be time consuming and also it will be error. 0.5 mm is okay. If you go for very, very sensitively, 0.2, 0.3, 0.4, etc., it will be time consuming and also practically it will be difficult. And that itself may cause error. So 5mm is the best one. You can go for the morphology characters next one. So when you take the meristic characters, meristic characters, it's got one very good plus point that is very good to merit because the meristic characters are fixed even in the birth itself. For example, when the larvae comes out, well, it has got the mayonnaise, it gets modified in the form of pins, If the characters are the meristic characters, whether you take the dorsal fin or anal fin, whatever it is, the characters are fixed at the right from the birth itself. So these meristic characters will be of very good use, understood, because there is no question of changing the meristic characters with regard to sex, with regard to length groups or like coloration. It may not change. So the meristic characters can be given importance even in our star assessment studies. Also, the meristic characters are given importance even at the delineation of the star. Understood, particularly the races. Understood. So the meristic characters are much important. And the counting of these meristic characters, all those things to our teachers might have been got in the next slide. Okay, now the another thing is the cross conformity. Of course, this needs a separate a lecture, so to say. This is one of the in the last 20 or 30 decades, the trust studies has been coming into existence because you go for taking the whole picture of the fish in a geometric pattern. Unlike the conventional morphology, the entire body is taken into consideration. So, even for the closely related World Taxonomy, the trust morphometric characters are very good useful. And further, go to the next slide. Further, if you take our conventional morphometry, which of course now it is also in vogue, and previously also it is so. Just we take go to the next slide. You take this sort of measurements. Everything is two-dimensional. So everything is two-dimensional. So the entire body of the fish is not fixed as a whole. Go to the next slide. I think that figure so if you go to the trust morphometry, well, I think the figure is shown, but it's not there. Okay, no problem. The entire image of the fish is taken, unlike that of the previous. Unlike the two-dimensional morphometry, and in the two-dimensional morphometry, we take the characters either in the form of fork length or standard length or total length, and these are the three attributes. We take it as an independent variable, and the other characters, the head length or some 25 characters are there. Interorbital with snout length, snout to first class, etc., etc., etc. They are all taken as a dependent variable. And from the two-dimensional morphometric studies, go to the previous slide. Then we go for the it is always better to take the fork length if the caudal fin is typically forked. If the caudal fin is not typically forked, I advise you can take the total length. And better to avoid the standard length. Of course, for some of the fruited fish, particularly in the case of the sardinella and also in Anchois and also in the case of the Engralis like Risai, well, the standard length will be good. But for everything, the standard length, we have to take it in the sunlight and we have to see the end of the hyperal plate. Because it starts from the snout to that of the end of the hyperal plate, So every time we have to see the hyperal plate. Hyperal plate is a fixed for the last column, 3 or 4. Understood. This is a little bit problem in the case of the standard length. So better to take the fork length if it is typically fork fish like that of the carangids or lepharinids or limitids, whatever it is. Understood, or better to go for the total length. And of course, as I told you, this is not a image capturing. The two-dimensional conventional morphometry is not an image capturing. Just we use the range of ratio, mean ratio, standard deviation with the percentage of overlapping and also the percentage of non-overlapping for different species comparison, then we are evaluating. We taxonomists take if the percentage of overlapping is less than 5%, we take it as not a valid character, but a reliable, a dependent, so-called valid character in quotation, but a reliable character. That's all, not as a diagnostic key character. Understood. And remember, we come to this conclusion because not with the 2 or 3 specimens, we come with more than 25 or 30 specimens covering with the different groups and different sexes. Then only we are coming to the point that it is a reliable character, and when you add the diagnostic characters, the Stoutland could be distinguished between Thyrsa malabarica and Thyrsa cetracis because the overlapping is at 3.5% or 2.5% level. That is based on much amount of more number of specimens. So this approach we used to do, but now this approach has totally gone. We used to draw it in the dice diagram of ups and ups, where with the standard deviation, mean, range, overlapping ratio with 95% confidence interval. And this aspect is now totally gone. Now we have gone for the trust-morphometric characters, which is where we use 3 or 4 types of softwares, particularly the TPS4, TPS6, and TPS15. Then upon using this, we used to go for very statistical package like the principal component analysis, discriminant function analysis, cluster analysis, and also we go for the factor analysis. And from this, we go for the variable derivation, and from the variables, we will mark which variable is significant and which is insignificant. So this is a very good study that was morphometric, and as to even it could be done at the lower taxon level, with below the subspecies or below the what you call it as races or stocks, something like that. Understood. Next. Then another important study is the osteology. Of course, when I tell osteology, some of the, even I told my students, understood, why can't you, last semester, she is sitting here, understood, why can't you come with a heater and with the enamel, this thing, and 5 or 6 specimens to my room so that I can boil and I will show all the bones, etc., etc. They said, okay, but nothing came out, understood. Because the osteology, I will not, as a taxonomist, I should not call it as a boring one, understood. This has to be done. This should be known, understood. With a very good camera lucida, understood. With a good Alizarian staining preparation, and even I advise the youngsters of the Department of Fishery Biology, understood. You go for the Alizarian staining, give the preparation of the Alizarian staining also, understood. The Alizarian staining is the best one. Everything right from the head up to the last cordon vertebra, it will be very beautiful in gold and yellow color. If you preserve it, it will give an excellent lookage. But it comes only by trial and error practice. Sometimes, all the bones will break. Understood. The concentration, you have to select it in such a way. It is a very good storage medium. Understood. That could also be done with the Alizarian staining preparation in osteology. Now, coming to this osteology aspect, is you have got different bones, Well, right from the frontal to that of the last one, exoccipital, and all the vertebral bones, and also the ootic series. Understood. Epiotic, opisthetic, spherotic, erotic, and all the ootic series, Well, the young lads, maybe they find it a little bit difficult, and not difficult. They will not be that much interested. Understood. When you compare with that of molecular characters like mitochondrial DNA, etc., etc. So this osteology is a very good useful, particularly in differentiating at the higher taxonomy. Remember, the meristic characters, the morphological characters, the morphometric characters are good, useful both at the higher taxon as well as at the lower taxon. But as far as the osteology is concerned, only the bonal variations, because that could be useful, particularly at the higher taxon, because with the help of a single vertebral bone with the infracentral groove, J.J. Thomson was able to distinguish the entire family Mucilidae. Mucilidae, it is a challenging groove. Understood. If you get the Mucilcephalus, you cannot identify easily at the larval stage, juvenile stage, and the adult stage. really confusing because if a taxonomist wants to make a breakthrough, he has to take either Cyanide or Gilding. Understood? Because such a challenging task, And remember, in this osteology, with a single vertebra, Thomson was able to delineate most of the species. Understood? So the osteology studies will be of good use, but only the thing is, it would be of higher use, particularly in differentiating at the generic taxon or even at the above the general level. Understood? But if you have a very good catalyst and the boiling techniques, and you can also use the shape, the size of the bones, understood, for the delineation of the species. So this is one important aspect. And like go to the previous slide, and like your conventional morphometry, you can measure the cranial length, rostral length, diameter of the orbit, with depth of all the bones, and you can find the overlapping ratios, etc. And you can go for the delineation of the species. Next. Yeah, this is the training I have given. Understood. Interested persons can see how the fish has been totally bleached and see the coloration. Understood. If you put it in the jaws, it will be excellent colors. Understood. And ulcer staining is a good one. Next one. Next slide. This is a preparation. Next one. And these are all the bones, you have to boil, you have to take the entire skull out right from the over. So that is the exoccipiter. You have to carefully disarticulate and you have to wash it. And if more of yellowness is there, you have to add hydrogen peroxide. Understood. H2O2, then you have to go for bleaching. So if you put too much H2O2, the bones will disintegrate. So it all comes in trial and error practice. Then you have to draw very good diagrams. Understood. Using the diagrams. So these are all the various types of bones, which I will not go in detail because this itself is one of the next one. Then this is another important aspect. This also I do not want to go in detail. Understood. This also it may take more than an hour, but this as an information I am passing on to you. This is automatic chemistry, particularly stock delineation. Right from the 1990s, particularly for the temperate fishes like cod, salmon. Understood. This automatic chemistry is widely used because previously used to take the automatic Sagittar, particularly in the identification of the Cyanidae, I told the family Cyanidae is a challenging group. Understood. Because if you take the genus Autolycus, Autolycus, then Jonius, then Parajonius, all these cyanids, if you see it in the landing center, they will be more or less alike. Understood? You will be in a dilemma. So what we used to do, we will take the Autolycus, and of course, the Autolycus is very big in family Cyanidae and also the catfish Eridae. So we call the genus itself Autolycus and Autolycoides because of even for the young students, to take the autolyth in a Carangida or in a Lethrinids, it is very difficult because you have to sit and you have to break the protein bone, and inside the protein bone there will be three shells. One is Sagittarius will be large, another is Astragus will be large, and another is Lapillus will be large. You have to take only the sagittal bone, and the sagittal bones has to be removed, and that has to be studied. So in the case of minnows or barbers or in freshwater fellows, and also in the case of some of the marine fishes, taking the autolyth is difficult. So for the youngsters, if it is very big, they will be more fascinated. So for that case, we use only the arids and also the cyanids because the autolyth will be much big, Now this autolyth chemistry, is also gaining much importance, particularly in the taxonomic side. Next slide. So it is now particularly used to identify to delineate the fish stock. It is a very powerful tool. The chemical signature of the autolyths, understood. It's subjected to vary depending upon the environment and also depending upon the food in the particular aquatic system. So it has gone for the delineation of the stock scientifically, and of course, once we go for the delineation of the stock, because our multi-species, they are much based on what we call it as multi-stock oriented, unlike the single stock as in the case in the temperate fishes, So for fishery management purposes, well, this new technological approach, particularly the automatic chemistry, is of much useful, right? So for the stock concept, it is very good use to understand. And next, particularly for the this is applications not only the chemistry, particularly the physical-chemical properties of the surrounding environment will produce a distinct not only the chemical composition. We have to go for the evaluation of chemical signatures, not only, and this could be used, particularly using the X-ray spectrophotometry, and this will give the entire information of the lifespan of the individual. Sometimes, this chemical signature may also subjected to vary with the ontogenetic and also the environmental factors. And this is the next approach in the classical taxonomy. You call it as a soft anatomy. Well, up to the phylogenetic, the intestinal convolutions, the bladder, understood. For example, as I told you in the case of the cyanid fishes, the air bladder plays an important role, particularly in the identification of some of the challenging genera like Autodictys, Paranibia, Rivia, etc. The dissecting the otolith is of very good use. But remember, when you study the otolith, don't go to the landing center, and from the landing center, and you take the fish, and if you take good number of fish, don't put it in the formalin level sea, because we did it like that in our days. Understood? If you put it in formalin because you are so tired, so if you have gone for the night landing center from 11 to 3, you will be standing. If you come to the college, you don't put it in the formaldehyde, because if you put it in the formaldehyde, the entire air bladder will be out, the entire phylogenetic will be out, they all will be split. Understood? Your entire collection will be lost. So kindly you put it in the deep freezer, or kindly sit. At that time itself, then evaluate all the phylogenetic numbers, phylogenetic branches, and all those things, even in the case of the air bladder. This is one information, because this has to be known, because you simply go to landing center because of your overtiredness, you put it in the formaldehyde and you escape. So that after 2 or 3 days we can study. That should not take place, particularly in the soft anatomy, even with regard to the intestinal convolutions, So this air bladder, the phylogenetic, they are all important characters as far as the soft anatomical part is concerned. So whenever you want to go for a molecular, now let me go on to the another aspect, that is the identification of these commercially important families. Now, so to say, my conclusion is this classical taxonomical approach is, well, particularly for the researchers, understood. You can do it on 3 aspects. Point 1, you take a big genus, for example, genus Sardinella, or genus Lathrinus, or genus Lajanus, which is having more than 15 or 20 species, or genus Caranx or Carangoides. And since if it is a PhD dissertation, understood. Time is also a factor, degree is also a factor, understood. Was sleeping. Degree is also important, understood. So you go for exact delineation. For example, what I advise you is, so as usual, you want to study from the Gulf of Mannar. Understood. You take it right from Pamban to Veerapandi, Trichandur, or up to Kolachan, whatever it is. You are taking Sardinella, which is having 15 species lesser sardines. Understood. You go for the exact classical taxonomical approach, as I told you, right from the morphology, last end to the soft anatomy, including your plus morphology. Understood. For all the 15 species under the genus Sardinella, this can be a very good approach. Understood. If time permits, understood. Remember, you take good number of specimens, you die in the landing center, understood. You get more number of specimens, more length groups, more sex, then only the information which you publish it in the form of a monograph will be worshipped by many of the scientists, at least from the Indian side, understood. Otherwise, it will be thrown into the dustbin, it will be kept only in the library, understood. So, take good number of specimens. Evaluation should be there, which the methodology, of course, this I told it very in brief. And if you want it in detail, you come to me. I will tell it in detail, so to say. You go for excellent evaluation for the 15 species of the genus Ahilan. In the Gulf of Mannar coast, it would be excellent, including the osteology. This part. Now, since you want to make an integrated approach, you jump also into the molecular approach. Of course, one of my students will be giving the second or the third lecture. Understood. That could also be attempted. Understood. This integration will be the best. Point number one. And if you want to make a coastal comparison, East Coast and West Coast comparison, that could also be done for taking the entire genus Ahilan in the Kerala coast or in the Tamil Nadu coast, and you can use the crust morphometry and you can identify whether any delineation will start. And of course, not only chemistry, understood. Of course, if this subject is in the stage of infancy, that could also be used. So this type of approach, taking only the classical taxonomical approach, putting it in detail is one aspect. Understood. That is the best aspect, particularly in going for the delineation and the erection of a new genera or a new species. For new genera, new species, you follow all the principles envisaged in the IC, which I am not going to take into. So now this aspect, one can do it. Understood. You note down the colors immediately in the landing center. After the 1 day, coloration, putting it in the formula. After 40, everything you record. Understood. So that will be a very good approach, particularly for some of the fishes which shows variation in the coloration pattern. So this could be a best approach in the classical taxonomy. Now, coming to the second aspect, the identification of these commercially important families. Understood. Particularly the most important order, as I told you, there are only important orders. All are important, but from the biodiversity perspective, the Perciformis, but now the Perciformis itself is coming into many orders. That is a different case. So the previous Perciformis, which includes the 55 families, understood, considered to be one of the commercially important groups. And now, for example, mothers are based in Piliformis, and likewise, even in the Karangids are placed in Karangiformis. Understood. So many formists have come, and but if you take the general purview of Perciformis, so the first, because if you go to any landing center, because I am putting it as far as in a brief situation, at the same time I want to enrich you with some points, at the same time taking the time also into consideration. Understood. If you go to any landing center, fish landing center, you will be 50% of the Perciformis group will be either Venkataramani, understood, or it will be Lagianidae, or it will be Limictridae, or it will be Hemulidae, or it will be Suranidae, or sometimes it will be Karangidae. Understood. These are the most important group, You can see at least 30 to 35% in a trawler. Understood. So, how to identify this commercially important Perciform families? Perciform in quotation, because some of the families are now put in orders. Okay. The first and the foremost thing is, I told you the importance of the external morphological characters. The first one is the case of the Lagianidae. Now, go to the next. For example, with the head region alone, you can identify some of the commercially important Perciform groups. We call Latherinus complex, Latherinus Largianus complex. We use the term because certain families are inserted in this complex. Understood. And all the southern families are commercially important groups, and you cannot very easily identify them in the field immediately after seeing. So you have to take the specimen and you have to have a good look. Then only you can delineate these families understood very easily. Of which the first is taking the head region alone and the presence of the scales where one could identify different important families of these Latinus Latherinus compounds. For example, you go to this. See, the presence of scales, which is totally absent in the case of the Latinus region. You can see 3 rows of scales. Understood? 3 suborbital region. So that is the best character in distinguishing 1. Lagianidae, 2. Nimitridae, 3. Hemulidae, 4. Serranidae, 5. Carangidae, then 6. The important group is the important group inside that is the parrotfishes, scaled. Understood? Now, we go to the next one. You take the Lathyrus. You see the scaleless patches. To have the scaleless parts in the Lagianidae, which I showed in the slide. Then, you take 3 rows of scales, 3 about. You take the big face bleeds. Lathyrinids are having the extreme common look. One is the big face bleeds, the other is the emperors. So one is big face, the other is the harmless emperors. Understood? So you see the absolutely no scales. This is a characteristic feature of Lathyrinids. Hemorrhoids. Previously, in our times, in 70s and 80s, hemorrhoids were called as homodicity. Understood? But under the law of priority, hemolyt, genus hemolyt takes its precedence. Understood? Now the family has been changed it to Hemulidae, the homodicidae. You can see in the World Taxonomist Appreciation or the Fisheries Science by J.S. Monroe or Indo-Australian by Leader and before the homodicidae is changed. Now you see the scale pattern in the Hemulidae. Understood. Now you recollect all the important families, how the scaleless patches it distinguishes this important, commercially important families. Why I have taken these families? Because today, if you go to the landing market, at least 30 to 35% will be taking one of these scales. Besides families. So next. Really ceramics. The presence of 3 spines and the maxilla being totally exposed, which is the character. Understood? You cannot see in the other families which I have put. So, with this we will stop. We go to the first one, Lathyrinidae. Not Lathyrinidae, the first one, Lagianidae. See, if you take the Lagianidae, family character too. Now, here next one. This Pinjalo, next one. The Sassiuridae, understood. They are all the groups which are very, very closely related to the family Lagianidae. So, what Fisher and Bryan said in his FAO identification is, and they have given, if you draw a straight line, understood? This straight line, it should scout close. It should cut the center of the caudal fin. that is taken as the character for distinguishing the Sassyonics with that of the Latvians. Understood. Just take a cross. Understood. And apart from that, unlike the Lagenics, the Sassyonics, they will have the fusiform bodies, unlike the rhomboid bodies in the case of the Lagenics. Further, the later line states will also be very, very prominent, particularly in the case of Lagenics compared to Sassyonics. And in the case of the Newtons, we have two. One is the genus Newtons, where there's prominent spine is present in the case of the genus Collapsis, and not so, go to the next slide, and not so in the case of the Newtons. And further, you can see, and further, the caudal fin of the genus Newtons, the upper caudal fin is developed in form of a small big fillet. That is one of the characters for the identification of the genus. And of course, the Thoothukudi is a very challenging group, Well, at the family level, you can identify very easily with the cheek, the scaleless, and also with the tail spines. Understood. Remember, the depth of the suborbital is very high, unlike the other closely related subordinates like the Jayakumar. We have got different area. One is the monotaxis. This is Hermodidae. Now, in the case of the Hermodidae, see, the Hermodidae could be very easily distinguished from that of the Lagenidae or Newtonidae or Serranidae or Lathanidae by the presence of pores or the GPs. Remember, pores are also present in the family Cyanidae. Understood. But in the case of Hermodidae, the lateral line, it will not extend up to the tip of the caudal fin. In the case of the Hermodidae, but in the case of the Cyanidae, the lateral line extends up to the tip of the caudal. So the presence of pores is a characteristic feature of the family Cyanidae and also the Hermodidae. But the lateral line, it stands apart. You go to the previous slide. And previous one. The gill arrangements is also very stubby, unlike the other closely related families. You have got very dark-like gill rakers. Remember, in Lathanidae also, understood. The gill rakers are very strong, but unlike in the case of these Hermodidae, we can very easily distinguish this knot-like gill raker, which is a characteristic feature of the Hermodidae. Ceramics could be very easily identified with the presence of small spines in the upper region and exposure of the maxilla. Not of time and we go to the previous. Next. The pharyngeals, of course, this is a very big family. Now it has been got in the form of an order pharyngeal party, and the best characters, of course, 4 characters. The lateral line scutes, of course, that is also absent. Other genera like Stoporoides and Brachylophus. Lateral line scute is 1 character, but is also absent in other genera. The second is the presence of 2 anal spines. Understood. Then the third is the concave pectoral fin, city-shaped pectoral fin.
And last is the pelvic fin, which is with 1 to 5 days. With these 5 characters below, it goes with the concept of this polythetic taxa concept. What is polythetic? Of the 5 taxonomistic characters, 3 characters will be present in closely related form and 2 characters will be absent. And 2 characters will be present in the closely related form and 3 characters will be absent. Overall, if you take all the 5 characters, then you can go for the clear-cut delineation, either at the genus level or at the species level. This we call it as the concept of polythetic taxa. Understood? When we go for the total evaluation of the characters, you cannot come to the conclusion all the characters will be present only in the particular genus. It need not be. Understood? Sometimes majority of the characters may be present in some of the genera and some may be absent. When you take the totality of the characters into consideration, then the concept of polythetic taxa works out. That is the reason only the letter line skews. The entire letter line is with the skews in the case of Trachurus, which is not available in Indian waters. It is a temperate species. A part of the letter line is with skewed, particularly the horizontal part. That is a common characteristic feature of some of the carangids. The entire letter line is absent without skews. That is in the case of these Combraides. You can go to the next slide. This one. Trachurus also, the skew is totally absent. If you take the Pampanos, then next slide. And if you take the Demopterus, the skews will be prominent, but that could be very easily identified as the presence of 2 figures. In the case of the Elevatus Viprillatus, understood, well, you will be getting the fingerets also, but if the body will be both producible. But if you take all these characters which I told the letter line skew is absent, but in all, the petrol fin will be in city shape. There will be 2 anal spines, and the pelvic is 1,5. Understood? Next. These are all the various genera which I need not go in detail. Okay. Okay, next one. Then Alicus. Next. Next. Then, of course, this is very easy. Family, which is very easy. If you take you can also make a comparison. Archiceptidae, Carantidae, Leoparidae, Scombridae. You can go for molecular taxonomy, and you can go for the phyletic relationship, because they tell these 4 families are closely related biochemically. Understood? The osteological features are also same in Carantids, Scombrids, and also in the case of Leoparidae. Understood? So, you go to the previous slide. You take the Scombrides. The entire body structure will be more or less similar to that of the Scombrids. What you call it as your Sheila, understood? Maybe. Understood. And osteologically, they have control. By the Japanese workers, these 4 families are closely related. But to my knowledge, I have seen no in-depth molecular taxonomy on these 4 families. And that may be a best study. Oleochromates, that means they identify the 3 genera. Oleochromates, the nature of the producible node. Oleochromates, Gaza, and you go to the next Gaza, then Sequita, then go to the next. Then we go to the side. Yes. Next. Of course, Moulitharan could be very easily identified to the presence of carbons, both opinions and also para-opinions. Next. Next. From carnivorous groups. For the limits, the presence of the pre-factor fermentates,
Next. Would be very easy. This yes. Now coming to the cyanidae. Cyanidae is a popular group. You popularly called as the croakers and drummers because they are called as the croakers and drummers because they have got a powerful solidification. Because it is sex specific solidification. Understood? So they are popularly called as croakers and drummers. Even for the port display, also for carrying, the sound specific, the sex specific sound is evident. So the sonorific puzzle is taken as one of the important identifying character in identifying some of the genetic taxon of the family side. Understood? And next slide. These are all the characters I have told. And with regard to the cyanidae, another important feature is, of course, your teachers might have told superior mouth, inferior mouth, and terminal mouth, all those things. Your undergraduate teachers might have told. But with regard to the family cyanidae, the food spectrum, the feeding habits are understood. Has also got a remarkable telling effect with regard to the species. For example, if you take Protonibia and Nibia, it has got superior mouth. So most the closest about it. And if you put Jonius and Autolycus, it comes to the midwater. So the position of the mouth is after. And if you take some of the genera such as Paranibia, where the mouth is terminal, so it feeds on the planet. So we take the shape of the mouth in the entire family cyanidae, and we go for the delineation of the generic taxon. Remember, cyanidae is a very challenging task and the species taxon. Family taxon very easily click. Lateral line extending up to the tip of the caudal fin. Sons of force on the chin. We throw it as a cyanidae. Whether it is Autolycus or Jonius, etc., etc. But if you take it, when you go for the species and subspecies level, then the entire subspecies, all the subspecies of the particular genus we look like. So for that, we go for the swim bladder, pyloric sweeting, or the cell. These are taken as a three valid characters in identity. Of course, if we are totally to be very easily identified, only for one genera, understood. The identification of the side is easy. Because in the presence of force, mental force, then you have got the bottom force, understood. Then subterminal mouth, in the case of the here, subterminal, it goes to that of the column feeding, right? And inferior mouth is the voyage of all these family cyanidae. A very interesting group, particularly if you want to correlate the feeding habits with regard to the mouth of the entire family cyanidae. The position of the mouth that also supports taxonomical characters. The proof of the mouth. Now you can see the presence of force, and another thing is variety of caudal fin is present. Eparginate, truncate, rounded, S-shaped, rhomboidal, but also point. Understood. Endrigium. Understood. Terminal, subterminal, inferior, superior, all those things. So all this coincides with the mode of its habitat. Understood. Then the presence of force in the lateral line extending is a close for the character. Then the swim bladder that is taken as an identification character for evaluation species taxon. Next one. Next. There are not only TSOs, cyanidae, lot of type. Of course, how to cut out cyanidae, you will just write down for you. Next one. Next. Go quick. Go to the previous slide. Automic cyanidae I have shown with the view of the head from the top. The otolith, the shape of the otolith, particularly the inner view and outer view, shows a remarkable variation, particularly the identification of the species taxon of the particular genus. Next slide.
Next, 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 next. Then coming to the family Stombridae, this is also one of the, now Stombridae is also taken as a different order, and Pullets, which previously included in the family Stombridae, now it has coming in a separate order, Tuliformes, next one. Very, next, go to the figure. Figure part, this could be very easily identified at the family level because you have got both the spinals, first dorsals and secondary, and also you have got many finlets. Apart from that, coupled with it, you have got the caudal keels. Now, caudal keels are present in another ornamental group, that is Echinoidea. Popularly, they are called as the surgeon fishes. Because why they are called as the surgeon fishes? Because the caudal connecting keels is poisonous neurotoxin. Understood. But it could be very easily distinguished that the antherids can be orally fishes. It could be very easily distinguished from our strawberries. There are top carnivores like the commercial department, and secondary carnivores are also like tunates or important food fishes, could be very easily distinguished not only by the presence of the keels, but the second dorsal feelings given in the form of it. That's next one. Very commercially important families of its next. Next. You will ask, another order is the important is also I will rush it because the entire topic could be taken for 3 or 4 hours. That will go to the next slide. Of course, you know the characters of the topics which your teachers write on not single dorsal. No face fight that can be well behind the technology order for the topics. Next. So, in the case of the family Groupidae, if you take the commercially important genus, you have got this is the entire diagrammatic sketch of the groupioid fish. Well, first is the belly tube. Is a characteristic feature of the family Groupidae. In some, like Ablagaster, it is absent. Understood. We have got 2 maxilla. Go to the figure is understood. Then, well, in some, you have got a post-pelvic tube also. Of course, single dorsal feet is a characteristic feature of the family. Understood. Then, if you want to identify a particular genus that is studied in the Ablagaster, but also in Indonesia and Pelora, all the Gemina writing of which I am not very patient with. All are covered in important families. Understood. The frontoparietal stray in the head region. See the head. See the head region, the real part of it. The frontoparietal stray gives a very good valid characterization in identifying the genus Sardinilla, the Amblygaster. Previously, Amblygaster was included in the genus Sardinilla. Now, Amblygaster serum is a closely related species part of the genus Sardinilla that could be identified. I will show you the figure. The scales, the lateral line, are uniform on that, whereas in the case of the Sardinilla, it is either largiceps or tissue or pegasoma or degree, whatever it is, all the are. Now, when you take this, what is frontoparietal stray for Pelona, for Indonesia, for Antagaster, like for Sardinia, the frontoparietal stray, the head region, could be the variations, could be very easily identified. That could be taken as a valid character because the Groupiformes, if you go to study the molecular characterization of the entire Pediaskin orders, when the Groupiformes are supposed to be the order, understood, which are said to be primitive, compared to the most specialized order, you call it as Siluriformes. Understood. Perciformes is also a little bit degenerative order. Understood. The specialized one you go for the Silurids, the areas. Understood. But also some of the specialized group like Scorpioniformes, understood. So if you take these Groupiformes and Basic Groupiformes, They are all in the evolutionary ladder. Of course, if you go study the molecular characterization, it may also throw more light, I presume. Understood. These are all the groups, understood, which are said to be very, very primitive. Understood. The primitiveness stands around are the presence of the scutes on the belly region, but also they have got a single dorsal fin. Of course, two dorsal fin, three dorsal fin, they are all said to be predictive. But a single dorsal fin or two with the ray is, see, when you want to study the evolution, there are three types: primitive, advanced, specialized. If you take the flatfishes, they come under the specialized group. If you take the persicorps, it comes under the toothpicks, they come under the primitive group. Advanced groups, and so is the case with the Latifera, there is our group with the cylinder showing. And if you take the advanced characters, well, some of the groups like dipnod, fishes, these comes under the advanced group. So we go for, according to the habitat, the characters will be specialized. Understood. So if we take these basic cruciforms and cruciforms, they are the primitive groups, and they are coupled with many groups and single dorsal. And you have got this area, characteristical case, is what are the valid character in distinguishing the other genera, that is next time. Spectrolidus, 48, those groups in code, they get Sardinilla is one of the important genera. They very easily identified from the other genera with the presence of both the characteristical case and skewed present. And also, you have got the important characters that is supervised. This is the protoparent X-ray, which I showed you on the top of the head. It will be very easily identified. Then the presence of this arrangement of the second superbacteria in heterocyclics and Sardinilla. Then parts of the upper, this is taken as an important character distinguishing mechanism. Then Aplagasta, as I told you, the three dorsal speed forming magnifying single medium probe attached to the Sardinilla, by which the genus has been this species setup has been removed from the Sardinilla, and a new genetic status is given to Espinosa. The body form itself is what the Ilsha again. Ornamental right? It's Nebulosa. Nebulosa very easily identified by the presence of the last of the element. These are the best characteristics. Very long and like all the other genera family less than Opisopteris, they are morphological changes or the erecting. Then commission is the granite well, we have got 3 more genera, that is Moulitharan, Fisheries Science, Ichthyotaxonomy, endemic, and also Suresh. Coming under the order, fifth one is the Durairaja is long. What is it? You take the Durairaja and it will come down and walk. It will not be that much strong. The next time, this is failure where the body shape is different. This is very depends on the minutes. Now, coming to now, this is a very important group. Entomology fishes, popularly called as adpolites. Next one. Now, introduction of policies. Diagnostic characters. Well, the first character goes to the presence of species. Some genera, if you take the
Stolliferous and Chrysocolira. If you take the preside, we have the first also the big. And apart from that, the lower jaw will be extended beyond that. And in the case of the preside, the lower jaw, might be extended. And in all the family, on all the species in the family of Cryptic, the lower jaw is not. Next one. Next slide, please. Because bigger I do expect next one. Now, recently the Empressicolida genus has been erected. Previously, all the species of the genus Empressicolida is based with the genus. Now, the exposure of the hero whale, understood, is one of the valid characters, particularly differentiating the genus Empressicolida and Stolliferous. And also the Isthmus is this region, understood, is reaching up to the tip in the case of Empressicolida and not so in the case of the Stolliferous, by which these two closely related genus are distinguished. Of course, Coelia is a temporary called erecting. So, if you take the Empressicolida Stolliferous, we have got only the pre-pelvic species. And if you take the Presa genus, it has got pre-pelvic and post-pelvic scutes. And in the case of the Centipede, apart from the morphological structure of the body, we have the second. So, for maxillaries, next one is the spleen. The extension of the maxillaries, and which all these intelligence. This is another important group. Very, very brief. That is the chromatic of plant fishes. It's also important group. Next one, we have got next one. We have got 6 families. Understood. Bothinae. So all the 5 families to be very easily identified in the order chromatic formation. 1. With the edge of the upper pole, either it is or 2. Ice may be present on the left side or right side, sometimes the ice may be present on the right. 3rd, the origin of the dorsal just in the middle of the eye or after the 4th is the spinous nature will be the 5th character is confluent nature of dorsal order again. So, all these characters you take into your mind. With these characters, you can identify 1. Go to the previous slide. Go to the previous. You can identify Bothinae. Okay, next one. Similarly, you have got eyes both on the left side and right side, but here the pelvic is one side. Understood. Unlike the other species. Then the iris is placed on the right side. This is the Bothinae. Next one. Cyanoprosits, go to the bigger part. All not pre-attention, very easily identified. Understood. Next one. Then right eye is called as a chronodelphidae. Next. Then Cephalidae, of course. Now it is also becoming very rare. Previously, we used to get lot of Cephalocephalidae. Now this particular species is orthodoxy. Previous time in 19th or 80s or 90s. So that could be very easily identified by the first dorsal, the dorsal fin starting after the eye, and also the presence of very powerful spike also, by which the Cephalocephalidae genus could be very easily identified. The soles, understood. Both the real side and the the soles, they have the eyes on the right side, but here the entire tree of maxillary region is exposed, unlike other family order for protective topics. It's a genetic policy reference for an order. Certain features I already told you, or looking bad states or hidden features that I don't want to go into yes, because I have taken that parent features, their butterfly features, so they take second. So last says, let us get the whole pattern, homosexuals, beliefs, varieties, the answers, characteristics, so this goes with the overall picture of the commercially important marine properties, of course. Each and everything we can go into detail, but due to the loss of time here, we are not told it in detail. But since more than 30% of this lecture your undergraduate teacher might have taught you, understood, and remaining understood, may not be due to you. And if you have any doubts or any question or any clarification, you can ask. Understood. So better to go for the identification using the particular as well as the classical taxonomic studies. So thank you for your great community. For those that are around and any doubts or any clarification, understood. You can very free to ask. I'm very sorry I have run with very much fast. Understood because there is no other option. Even I have not told about the siluriform groups. Understood. And also Bugilids. So, any doubts apart from that? There are some. Well, thank you.
So thank you very much, sir, for your elaborate and informative lecture. Within a short period of time, you could touch both classical taxonomy methods and then marine fish taxonomy. Thank you very much, sir. Students, let's have a break for 10 minutes. Tea and snacks are served outside. Please be back after 10 minutes. Those who are present online, you also please join after 10 minutes. Please don't take more than 10 minutes. So you have exam in the afternoon, so we will be winding up exactly.
Mauli, am I audible to you?
Yes, you are audible, sir.
Okay, are you ready?
மறுபடியும் எல்லாம் ஸ்லைட்ஸ் வச்சிருக்கலாமா?
Sir, இருக்கு சார்.
ஓகே மரை. இது உங்களுக்கு அடுத்து வந்து சுரேஷ் சார்.
ஓகே சார்.
நீங்க பார்த்து ஓரளவு ஃபுல்லா எடுத்துருங்க. சரியா?
ஷூர் சார்.
ஓகே மரை. பார்க்கறதுனால you can have more slides. I'll take a new area.
Very good morning. So now I invite the second speaker, Dr. N. Moulitharan, Adjunct Professor, Department of Fisheries, Resource Management, Dr. N. Fisheries College. So we specialized in freshwater fishes, particularly snakeheads, catfishes, and barbs. New species of fishes, 2 recovery collections, and few new records to India. So with these few words, I request Moulitharan to start his presentation. I also request the participants to mute the
Sir, my slides visible, sir?
Only it is visible.
Okay, sir. Shall I proceed, sir?
Yes, Mauli, it is visible.
So, good morning to everyone. So, on this day of occasion, the World Taxonomist Appreciation Day, I'm glad to present my presentation on the taxonomy of freshwater fishes, particularly the Indian region. So, my specialization is on freshwater. So, I deal with freshwaters in my PG, PhD, and my expertise in snakeheads. So, I'm starting with my lecture. So the importance of taxonomy. So taxonomy is the systematic arrangement of the components of biosphere in this earth. So every living material we have to arrange it, so classify it, so we have to so that we can understand. So the fishes are the products of the vertebrate evolution of 100 billion years of vertebrate evolution. So they are not just created by a day. They were the products of 100 billion years of vertebrate evolution. So they have ecological and economic importance. So for conservation and implementation of conservation programs, and we need fishes for every kind of from food to for our stability of our ecosystem, we need fishes. So for conserving these fishes, we need to know about the fish species. What is the exact diversity? Where do they live? So for that and all, we need taxonomy. The taxonomy is a base for everything. So the till date, today, the status is that many species are going extinct before even they are discovered. So many aquatic systems are being destroyed irreversibly. Urbanization and climate change, they forces the river to die. So many number of species are going extinct before they are discovered. So one such conservation, so the taxonomy is very essential and it is timely need. So we have to proceed much faster. Say for example, the knowledge of taxonomy is very essential in conservation point of view. For example, this happened in India, Tamil Nadu particularly. So we have Maharishis in Western Ghats. We have the Mahashis population in Kaveri and Bhavani rivers. So for a purpose of conservation, some Maharishis juveniles were brought from Lonavala Mahashir hatchery, and they were stocked in Bhavani river. So this happened in 1980s, 85 to 90s during the period. So they were thought that the mushy that occurring in the northern Western Ghats is the same species as occurring in the southern Western Ghats, but it was different. So what we have now is we have realized now only we have the Taur Ramadevi, which is endemic to Kaveri. So before that, before the realization of that truth, so people have stopped the Taur malabaricus looking like species. So that itself is a hybrid. People are saying that itself is a hybrid. So Taur malabaricus hybrid from northern Western Ghats, they are stocked in the southern Western Ghats. So the lack of taxonomic knowledge have created a genetic pollution here. So this is the impact. This is one of the kind of impacts which we face without the lack of knowledge without the knowledge of taxonomy. So the shortfalls, particularly taxonomy, we have certain shortfalls. The Linnaean shortfall, Wallacean shortfall, and Darwinian shortfall. So these shortfalls impact the taxonomy, the level of taxonomy, and the stage of taxonomy. For the first one is the Linnaean shortfall. So this shortfall deals about the formally described species and the number of species that actually exist. So this says that if say for example barbs, so we have 10 to 30 species of barbs in our river systems, but there may be about 40 to 50 species of barbs. So that is a lack, there is a lack of shortfall there. So the second one is violation shortfall, the lack of knowledge about geographical distribution of a species. So certain species are point endemic, certain species are region endemic. So for example, the Taur Ramadevi, which is endemic to southern Western Ghats of rivers of Kaveri. But initially, the people thought that all the Taur species occurring in the entire Western Ghats rivers are the same. So this is the shortfall that occurs in the distribution, exact distribution of each species. So this is called the Wallacean shortfall. Then the next one is the Darwinian shortfall. This says about the lack of knowledge in the tree of life and the evolutionary lineages. So next level of taxonomy, today we're dealing with molecular level of taxonomy and the evolutionary biology. So this shortfall says that we have to know about the evolutionary lineage to ascertain the taxonomic level of certain species. So we'll be seeing about these shortfalls and how to rectify these shortfalls in the future slides. So as this day is being the Taxonomist Appreciation Day, we need to know about some taxonomists who have created the foundation, who have laid the foundation of the taxonomy that is having we today. The taxonomy we have today is developed by many number of taxonomists, starting from Aristotle till today. So first few slides we'll be seeing about some taxonomists who have laid the foundation of the entire animal taxonomy in the earth. So first one starts with 300 BCE. The first one is Aristotle. Dr. Aristotle, he described 170 species from the Mediterranean Sea. Before that, even scientific science says that documentation literature says that Aristotle is the first one to start the animal taxonomy. But we have many number of our literatures. We have, while citing our Tamil literatures, we have thousands of years old literatures which documents and refers the species. For example, the oldest dynasty Pandyas, they have the fishes in their flags. So they've recognized the species and they have made it in their flags. So that is the important they have given to the fishes. So next to Aristotle, the early ethology based on the documentation, scientific documentation. So first one is to be Aristotle during 300 BCE to 1499 CE. Followed by the modern ichthyology starts with after the 16th century, Pierre Bellon and Guillaume Rondelot. They have described about maximum of more than 100 species. They have written their books on two books of aquatics and species marinum. So these are the books written by the initial taxonomists. Then comes the new era. The new era of taxonomy starts with the 17th century, where John Ray and Francis Willoughby, they have written a book called Historia Piscicum, in which they have developed a rational system for the classification of fishes. They have in which they have classified 400 number of species in their book. So these books are available. These books are till now we are available publicly, openly available for everyone in the Biodiversity Heritage Library. You can download it and we can read. Still, it is available there in the original copy itself. And then for the next, the Peter Attidy in 18th century. So he is the colleague of Carlos Linnaeus. He is called as the father of ichthyology. So he drowned at the age of 30. He died at the age of 30. He drowned, I think, drowned in river. So he developed the standard methods for making counts and measurements, which is the essential part of taxonomy. So Peter Attidy established the standard methods for making counts and measurements of fishes, which is used for taxonomy. So after the death of Peter Attidy, Carlos Linnaeus published his books, Bibliotheca Ichthyologica and Philosophica Ichthyologia, in 2 volumes, 2 books. That was written by Peter Attidy. And followed by Peter Carolus Linnaeus is called the father of modern taxonomy. He only introduced the binomial nomenclature. We have the two-word nomenclatures, the genus and species. So Carolus Linnaeus brought the system by a term called binomial nomenclature in his book Systema Naturae. So actually, Systema Naturae was published in 10 volumes. So after that, the student of Carolus Linnaeus, the Peter Tangbang, he has surveyed under including Ceylon. He has surveyed South Africa, South Africa, Japan, Ceylon, and he has documented many fish species. So after the 19th century, modern era taxonomy begins with the 19th century. Here we have Elsair Bloch. So Bloch is a German researcher. So Bloch and Schneider is also a German researcher. They used to receive samples from all over India, but from all over the world. They studied the specimens and classified the fishes from all over the world. But they have also dealt with Indian fishes. So there was a Dutch settlement in Tarangambadi. They supplied the materials from Tarangambadi to Bloch and Schneider. So we can see the many number of freshwater fishes named after described by Bloch and Schneider. So they received the materials from India with the help of Dutch settlements. And the Lacepede also have described a number of species from Indian waters. His book, he wrote his book Historinella Natura des Poisons. So this book also deals with many number of Indian fishes. The books written by Bloch in 1785, the Schneider in 1801, and Lacepede have dealt with many number of Indian freshwater fishes. The next one is Cuvier. So he is also has written a book called Histoire Naturelle des Poissons. This is a French book. Mostly the entire book is in French. We have to translate and we have to observe the facts while reading these books. So after the Cuvier death is the work of Cuvier was continued by Valenciennes. So this could be observed in many species names. We can see that after the species name, the authority will be Cuvier and Valenciennes. So Valenciennes and Cuvier have laid a foundation for the classification of many number of species, and that standard as example for many zoologists, future zoologists. And Jean-Jacques Dussumeri. So he is not a taxonomist, but he was a voyager. He traveled across China, India with his own ship, and he has collected materials from these regions, and they have supplied to Cuvier. Actually, Cuvier referred him as a young man who has already made several voyages. So he has referred in his book. So around 36 species has been named after Cuvier. So we can see named after Dussumeri. Cuvier named these species in recognition of Dussumeri. Around 36 species from India has been named after Cuvier due to his contribution, due to his material supplied for Cuvier. Then pure Indian ichthyology starts with Patrick Russell, Dr. Patrick Russell. So even though Lacepede, Bloch, and Schneider have dealt with some sort of Indian fishes, so Patrick Russell was the one who started to work exclusively on Indian fishes during 1803. He written a book on the fishes of Visakhapatnam, wherein he described 200 number of fish species. So Patrick Russell is a doctor. He is a surgeon, British surgeon. He covered the fishes from Cholamandalam coast, the region below Krishna River. Then the next comes Francis Hamilton or Buchanan Hamilton. So he dealt with the fishes of Ganges, and he wrote the book called An Account of Fishes Found in the River Ganges and its Branches in 1822. Still now, this book serves as one of the major reference for the fishes of North India. The many number of fishes which needs to be identified from the North Indian waters like Ganges or Brahmaputra or up to Krishna. We use this book as the standard reference for identifying the freshwater fishes. Followed by Hamilton, McClellan. Actually, McClellan collected the materials described by Hamilton, and he have also published the book called Indian Cyprinidae in 1839. Actually, Hamilton hasn't collected any materials. He did the description part. Followed by Hamilton, McClellan collected most number of fishes described by Hamilton, and he has published the book Indian Cyprinidae in 1839. So this book also serves as the standard reference till today. And followed by Theodore Cantor. He wrote a book on the Malayan fishes, catalog of Malayan fishes. After Cantor, we have the Bleecker. So he published the book called Ichthyotaxonomy of Bengal, where he dealt with many fishes of even the Ganges. He also written a book on Ichthyological Atlas of Dutch East Indies. So we in this book also many number of Indian freshwater fishes has been covered. and also many number of new species also been described. Then it comes the Francis Day. Dr. Francis Day was a British surgeon. He wrote the book The Fishes of Malabar in 1865, and the next book is The Fishes of India. Still now, this is one of the major. It is being considered as the Bible of Indian fish taxonomy or freshwater fish taxonomy. So he wrote the two books, only Fishes of Malabar and Fishes of India, being natural history of fishes known to inhabit the seas and freshwaters of India, Burma, Ceylon. So he covered a lot number of fishes up to 1,400 number of fish species. He have described many number of new species in that. Still now, for as Hamilton book serves as a standard reference for the North Indian fishes, Francis Day book serves as the main reference for South Indian freshwater fishes. So we are using till now we are using his book as the main reference for the freshwater fishes. And this was one of the remarkable work done till today by Indian fish taxonomy by Francis Day. And after that, because of his work, he was recognized, and later he became the Inspector General of Fisheries in India. And followed by Francis Day. And the next one is Gunther. Actually, Gunther worked on the museum's depositions. So all the earlier taxonomists used to collect the materials and describe it, and they deposit their specimens in British Museum or American Museum. So Gunther worked in the British Museum, where he published the Catalogue of Fishes of British Museum between 1859 to 17, multiple volumes. Actually, he described over 6,800 species. So his work is one of the monumental work where he solved many number of taxonomic confusions, because a single species has been described by multiple times by multiple researchers. So he solved that kind of synonymy and eliminated maximum number of confusions in that. So the work by Gunther was one of the remarkable in freshwater taxonomy, and the entire fish taxonomy. He has covered up to 7,000 species. And the next first Indian to be after Gunther, the first Indian to be play a major role in Indian ecology is Dr. Sundarlal Hora. He was the second director of Zoological Survey of India after Bani Prasad. So he wrote many theories called the biogeographic theory of affinities of Western Ghats and Indo-Malayan forms. So his theory states that how the fishes in Western Ghats and the Malayan region have the similarity. He also has Horace Satpura hypothesis. So these are the hypotheses deals with fish distribution and the affinities between the fishes in one Indian subcontinent and also the Malayan continents. So here, in his honor, we have a genus called Horabagras. Horabagras is a catfish genus which is endemic to Kerala. It has been named after him, and also a rice fish has been named after him. It's called the Hora ichthys. He is the first Indian known to play a major role in Indian ecology. Followed by Sundarlal Hora, we have A.G.K. Menon and K.G. Jairam. A.G.K. Menon further developed the Satpura hypothesis. Actually, Satpura hypothesis states about the distribution of fishes based on the landmass. And followed by Menon, we have K.C. Jairam. So K.C. Jairam was considered to be an authority of Indian catfishes. He wrote a book on Indian catfishes, and he also wrote a book on Handbook of Freshwater Fishes of India, Pakistan, Bangladesh, Burma, Sri Lanka. So this is also a major, till now it is one of the major reference book we used for freshwater fish taxonomy till today. And still this book has many number of flaws, which is being rectified nowadays. And followed by K.C. Jairam, we have Dr. E.G. Silas. We could have been known that he is the former director of CM of RI. He also play a major role in Indian ecology, freshwater fish taxonomy. And a catfish has been named after him, which is the Pungaceous silasy. Actually, this species is endemic to Krishna River. This Pungaceous species is endemic to Krishna River, which has been named after him, Pungaceous silasy. The next one after Silas, Dr. Ramadevi. Actually, Dr. Ramadevi is a botanist. She turned into be an ichthyologist, and she described many number of freshwater fishes from Tamil Nadu and also from other parts of South India. The iconic fish, the tar Ramadevi, which has been named after her. This is a fish which is purely endemic to Kaveri river systems, which we call it as the orange fin magashir, which is critically endangered now. Okay, so till now we've seen about the number of works done by various taxonomists from throughout the world and also from the Indian region. So now we're going to the concept of the taxonomy of freshwater fishes. So regarding to freshwater fishes, we can classify based on, say for example, consider Indian cichlids. So India has three native cichlids. India and Sri Lanka. We have three native cichlids. One is Heteroplus sudatensis, another one is Pseudotroplus macrotus, another one is Heteroplus canarensis. The Heteroplus canarensis is endemic to Western Ghats of Kerala. So other two species are distributed throughout in India. Say for example, take the Indian cichlids. So these species, three species are native to India. So here we can classify we can observe the fish with your visual eyes. So we can differentiate these species very easily based on their color and their morphology. So the first point of differentiation and recognition of fish species is by visual. So here we don't need much kind of morphological or morphometric methods to classify. So as they are visually or chromatically different, we can classify it based on our visual observations. So next stage, when some species are and some other species, like say for example the giant snakeheads. So these are the two species of snakeheads. They look alike, but they are two different species. So in the case of cichlids, they are, even though they are different species, they have very distinct color patterns and they also have different distinct morphology. So we can distinguish them based on the visual observations. But the next stage is the look-alike species. So they have similar color patterns, similar morphology. So in this situation, we have to go for the next stage of taxonomy. That is where we have to take for morphometry and merisics to compare with them. See, these two snakeheads are long considered to be the same species. The one is Channa micropeltis, this another one is Channa diplogramma. These two species look almost similar, but and they has been considered to be the same species. But later, the integrated taxonomic methods dealt by Dr. Venkataramani sir, the integrated taxonomic methods we use by the use of osteology, morphometry, and merisics. Later, we recognize that these two species are different. Actually, the Channa diplogramma is endemic to Kerala, and Channa micropeltis is endemic to Indonesia and Sri Lanka. But actually, before 5 years or 10 years, before the publication of this paper, Channa diplogramma was considered as the synonym of Channa micropeltis. So they were thought that there was a notion that micropeltis has a distribution in Malaysia and also in India. So with the help of integrated taxonomy methods, where they employed morphometry, merisics, and also the DNA, they have found out that these two species which look alike, but they are different species. So here, to differentiate the look-alike species, we are using the morphometry and merisics to classify the species. So the next one is the literature. So for marine fishes, we have FAO sheets and other kind of standard references. But for freshwater fishes, till now we don't have any dichotomous key. The key which serves as the exact to differentiate the two different species. But we have the situation is that we have to follow the standard literatures. As we seen the various literature by the earlier taxonomic researchers, the Hamilton's book and Bleecker and McLaren books can be used for the taxonomy of the gangetic fishes. The book written by Hamilton, Bleecker, and McLaren are particularly, they are reliable only for the gangetic fishes. And the resemblance of fish diversity, gangetic fish diversity. So what to say? The influence of gangetic fish fauna can be seen up to Krishna River. So the fishes which are occurring in Ganges can be seen up to Krishna to the major level. So up to Krishna waters, we can use the books written by Hamilton, Bleecker, and McLaren. So after Krishna, so the biodiversity changes, the fish pattern differs. So after Krishna, we have to use the literature written by Bloch and Snyder, Jordan, Francis Day, and Valenciennes, because these researchers have done their taxonomic work in here and they have published these papers. So for example, we have so we see the example in future slides. So what I'm trying to say is, based on the work where they have carried out, what they have published, which has to be used for the taxonomy of that particular region fishes. ஸோ எங்க வந்து இப்போ ஹாமில்டனும் ப்ளீக்கரும் மெக்லாண்டும் கேஞ்சடிக் ஃபிஷ்ல நார்த் இந்தியாவில் ஒர்க் பண்ணியிருக்காங்க. ஸோ அங்க இருக்க ஃபிஷஸ்க்கு மட்டும் தான் அந்த புக்ஸ் செட் ஆகும். ஸோ அதே மாதிரி கிருஷ்ணாக்கு கீழே அதே மாதிரி கேஞ்சஸ்ல இருக்க ஃபிஷஸ் வந்து கிருஷ்ணா வரைக்கும் they show the resemblance. So up to Krishna we can use these books, and below Krishna, but below Krishna we have to use the books written by Bloch, Jordan, Francis Day, and Valence, because they have carried out their research purely here. They don't have observed comparative metals from Ganges. So the next concept is species groups. So ஒரு species வந்து visually நம்மளால differentiate பண்ண முடியும் அப்படின்னா நம்ம we can done with visual observation. பார்க்கும்போதே நமக்கு difference இருக்கு அப்படின்னா we can do the taxonomy with, we can classify and recognize the species with our visual observation. So if the two species, ரெண்டு species வந்து ஒரே மாதிரி இருக்குன்னா then we have to go for morphology and morphomeristics. Then இதை தாண்டி cryptic species வரும், species groups and cryptic species. So these are the species, they have similar morphology and the morphometry, meristic characters. So they have similar morphology, morphometry and meristic characters which are overlapping. They call cryptic species. The cryptic species, the term cryptic species and species complex are used as same, but the species group is different. So these concepts, the concept of species group, species complex and cryptic species are constructed manually. So we construct these kind of concepts to solve the species confusions. For example, we have many number of species groups in each genus. I'll show in the future slides. So these species groups or species complex or cryptic species are more identical and they have the overlapping phenotypic characters. Overlapping phenotypic characters means the same morphology, same morphometry, same meristics. But they are different species with same meristics, morphometry and morphology. And they have wide distribution. ஒரே species ரொம்ப wide distributed இருக்கும், but எல்லாத்துக்கும் ஒரே scientific name தான் இருக்கும். They have wide distribution and called under one scientific name. And they have syntopic and sympatric. This is a term which describes that they have same habitat, ஒரே habitatல இருக்கும், but they are different species. ஒரே habitatல ஒரே மாதிரி இருக்கும், but they are different species. That is called cryptic species. For example, a genus can have both species group and also a species complex. So species group ன்றது ஒரு வேற, species complex ன்றது வேற. Both species group and species complex are the two different terms. So when coming to the species groups அப்படின்னா they are not essentially monophyletic. Monophyletic means they not have evolved from a common ancestor. They don't have ஒரே common ancestor இருந்து evolve ஆகி வந்திருக்காது. They have some phenotypical variations. But a species group can have multiple cryptic species. ஒரு species group கீழே நிறைய cryptic species உள்ள வரும். ஃபார் எக்ஸாம்பிள் சன்னா கச்சுவா அப்படின்றது ஒரு டிராப் ஸ்னேக், நம்ம எல்லாருக்குமே தெரியும். பட் சன்னா கச்சுவா ஸ்பீசிஸ் குரூப்னு ஒரு குரூப் இருக்கு. அந்த குரூப்புக்குள்ள இருக்க எல்லா டிராப் ஸ்னேக்கர்ஸ்ல எந்த டிராப் ஸ்னேக்கர்ல பெக்டோரல் பேண்ட்ஸ் இருக்கும். தே கால் சன்னா கச்சுவா ஸ்பீசிஸ் குரூப். ஃபார் எக்ஸாம்பிள் ஆல் த சன்னா கச்சுவா குரூப் that are distributed throughout India and Southeast Asia with pectoral bands. They do have pectoral bands. We can see the in the picture. So the pectoral fin has 4 to 5 bands. If any Gachua species have bands, they come under the group Channa Gachua species group. So this species group has many number of hidden diversity. Say for example, in the past decade, around 15 number of new species has been described from the Channa Gachua species group, but they look almost similar. So we can see the slide. இதுல 3 picture இருக்கு. 3 pictureலயே பாருங்க. Actually, original Channa Gachua. The first picture is the original Channa Gachua, described by Hamilton in 1822. The second picture is Channa Kalati, which was described in Sri Lanka. And the third picture is Channa Rara, which was described from the northern Western Ghats. So, but they are geographically they are isolated, but they look alike. So this is a example for species group. And also the Gara Mulaya is also one of the biggest species group. Gara Mulaya means நம்ம ரிவர்ல மீன்ஸ் ஹில் ஸ்ட்ரீம்ஸ்ல பார்க்கலாம். ஒன் ஆஃப் த ஃபிஷ் that used to stuck in the stones. That is also one of the biggest species complex in India, which I know which in a less number of work. The next one is the Channa Morleus. Actually, recently in 2020 they have discovered that there is a Channa Morleus species group in Sri Lanka. So they have, while doing the integrative taxonomy, they found that they have another species of Channa. One is Channa Morleus, another one is the Channa Ara. So both species look alike. பாத்தீங்கன்னா ரெண்டு ஸ்பீஷிஸ் பாத்தீங்கன்னா ஒரே மாதிரி இருக்கும். They ஒரே body pattern, color எல்லாமே they have the same kind of body pattern, color, morphology, morphometry, everything, but they are genetically different. So this is called species group, Channa Morleus species group. This paper was published in 2020 from Sri Lankan waters. Same also, India also has a Morales group. So Sri Lanka has a pure Channa Morales and also a lookalike Morales called Channa Ara, which is a separate species. Channa Ara. So next one is a species complex or a cryptic species. So in the all the cryptic species are monophyletic. On a species group they need not to be monophyletic, but they may be monophyletic. On a species cryptic species all the species in the cryptic are considered to be monophyletic. They evolved from the common ancestor. Or ancestor evolve but they where they look alike. Climate other distinguish பண்ண ஒரே விஷயம் means integrated taxonomy by using genetic distance and also other osteological features and other things. ஒரு cryptic species எடுத்து நம்ம resolve பண்றோம் அப்படின்னா we can brought about many number of new species. ஒரு species group உடைச்சாலும் we can brought about many number of new species. அதே மாதிரி when we break a cryptic species we can bring out many number of new species. For example Dawkinsia filamentosa, one of the biggest cryptic species in Indian waters. It is a bar mostly found in estuarine and low plain waters of Indian rivers. And another one is the Lepidophyllalis thermalis, commonly called spiny loach. Spiny loach is one of the biggest cryptic species in India and Sri Lanka, and also the filamentosa. So a recent paper published shows that the loach genus Lepidophyllalis in Sri Lanka and India, peninsular India, has many number of unrecognized and undiscovered new species. So they brought it as a species complex. The data from their paper suggests that Lepidophyllalis thermalis is a species complex in which multiple number of species remain to be investigated and described. So this thermalis species complex has many number of new species which has to be described. So this is a phylogenetic study they have carried out, and this is the picture right side இருக்குது பார்த்தீங்கன்னா உங்களுக்கு எல்லாமே thermalis தான், but see the color pattern variation in thermalis. This is from India and also from Sri Lanka. So they come under they have the same scientific name called Lepidophyllalis thermalis, but see the color variation, color pattern variation, and in left side you can see the genetic variation. So we can observe the many number of clades. So each clade can be a new species. So we have many number of new species under the name called under the one scientific name, Lepidophyllalis thermalis. So in the thermalis in the species complex, when we resolve, we can brought about many number of new species. So this is a species we have been suggested to be found as a state fish for Tamil Nadu. So this is the problem here: the lack of taxonomy and lack of. So if we describe, if we set thermalis as a state fish for Tamil Nadu, so future study may come. The species in the thermalis species, so-called thermalis species in Tamil Nadu was not the thermalis; it is a new species. So அந்த நேரத்துல நம்ம state fish name மாத்திர மாதிரி இருக்கும். So this is the importance of taxonomy here. So for ஒரு ஸ்பீசிஸ் காம்ப்ளெக்ஸ் இருக்க ஒரு ஃபிஷ்ஷை நம்ம ஸ்டேட் ஃபிஷ்ஷை வைக்கிறோம் அப்படின்னா அந்த ஸ்பீசிஸ் காம்ப்ளெக்ஸை ரிசால்வ் பண்ணும்போது அதுக்கான பேர் மாறும். அது நம்ம கிட்ட இருக்க ஸ்பீசிஸ் ஒரிஜினலா அப்படின்னு தெரியும். ஏன்னா இதுல என்னன்னா the thing is Lepidophyllalis thermalis has been described from Sri Lanka. It is not described from India. It was described from Sri Lanka. So it is native to Sri Lanka. So if at all original thermalis exists, it exists in Sri Lanka alone. So நம்ம இந்தியாவில் இருக்கிறது maybe new species கொடுக்கலாம், and many number of new species are available in this complex, and followed by the Dawkinsia bar pathipeng. This is a bar species which is commonly very common in ornamental industry, freshwater ornamental industry. So this is one of the large species complex in freshwater fishes. So recent paper has been published in 2020. So where they have resolved this complex, and they have described 3 new species from this complex. And இங்க என்ன பண்ணிருக்காங்க அப்படின்னா ஒரு ஸ்பீசிஸ் காம்ப்ளெக்ஸ் ரெண்டு ஸ்பீசிஸ் குரூப்பா ஃபார்ம் பண்ணிருக்காங்க. They have resolved a species complex based on the genetics, and they have formed 2 species groups called the assimilis groups and filamentosa group. So the top we can see the phylogenetic tree based on their genetic analysis in the left side. On the top red color இருக்கிறது filamentosa group, and the bottom blue color இருக்கிறது assimilis group. So they have classified a species complex into species groups. So இதெல்லாம் in the species complex, species group center they are constructed for our convenience to solve the taxonomy. So to solve the confusion, நம்மளே ஒரு group designate பண்ணிட்டு அதுல இருக்கிறத எடுத்து ஒவ்வொன்றா சால்வ் பண்றோம் to solve the confusion. Exactly. So these are the application based when we are becoming a future researchers and we are going to the work. So that time you will understand what is this. So next, so இப்போ வந்து நம்ம first we saw that if a fish with more number of morphological variations or chromatic variation, we can distinguish with visual observation. The next stage is when two species are look alike, we have to go for morphometry, meristics, and other kind of thing. So when the next stage is when fishes have similar morphometry, meristics, and color, everything, we have to go for barcoding and integrated taxonomy methods. So next stage is we have to use the biogeography and phylogeography. The next kind of methods we're using to do the taxonomy today. So recently we have a new species of snakehead has been discovered. It was called a subterranean snakehead from India, particularly from Kerala. It was a snakehead confined to the underground waters of Kerala. So they have discovered it as Animachana golem, a new genus, and later they have discovered it as a new family. So in the world we have only two genus of channa. One is Parachana, which is confined to Africa. The another one is channa, which is confined to Asia. So they have erected a new genus called Anigma channa, and they have erected a new family called Anigma chanidae. So this was the discovery in 2022. So after this, a paper has been published where they have done the biogeography and phylogeography of most air-breathing fishes in the world. So we can see the right top corner. These are the fishes, the air-breathing fishes of the world. They have done the phylogeography and biogeography by collecting samples, vast number of samples around the entire distribution. அதில் மேப் இருக்கு பார்த்தீங்கன்னா தெரியும். They have shown the worldwide distribution of each species. So in the மாதிரி they have carried out this phylogeography and biogeography, and they have found that the family erected as Anigma chanidae is invalid. So they have erected a family, new family, but the phylogeography and biogeographical studies have proven that the family Anigma chanidae is invalid. Rather, the genus may be valid. So நீங்க இதில் பார்க்கலாம். See the bottom phylogenetic graph. So where they see you can see can you able to see the cursor? Anigma channa golem is here below Parachana. So see, observe that and see the statement highlighted here. The description of a monogenic Anigma chanidae provides no information on phylogeny and only accompanies creating a group name that is redundant with Anigma channa. So what they have saying is the genus Anigma channa may be a valid one, but the family is not valid based on, because they come to this conclusion that based on the phylogeography and biogeography of the vast number of species, not only the single species, but they have carried out multiple number of air-breathing species. So science is proving and disproving. So they have here they have erected a family by using less number of comparative materials, and they have erected a family. But this paper was published in Nature, and following that, the other group of researchers, they have carried out vast sampling by collecting samples all over the world, and they have integrated phylogeography and biogeography, and they have disproven this disproven the family. So next to taxonomy அடுத்த கட்டமா இன்னைக்கு வந்து பயோஜியோகிராஃபி பைலோஜியோகிராஃபி யூஸ் பண்ணிட்டு இருக்கோம். The term biogeography, so see it and see the meaning. Actually biogeography means the actual distribution of a organism. Phylogeography means the lineage, genetic lineage, the distribution of a genetic lineage of an organism around the world. The taxonomy is going on the next level with the phylogeography and biogeography. So the thing is, applying species delimitation methods with spatially structured data will help to identify the species bound with geographic structures. So இதுதான் ஒரு ஆர்கானிசத்தோட பைலோஜியோகிராஃபியும் பயோஜியோகிராஃபியும் நம்ம எந்த அளவுக்கு எக்ஸாஸ்டிவா படிக்கிறோமோ, so that அதை வச்சு நம்ம we can come to conclusion that which is valid, which is not valid. And so this, up to this, the level of taxonomy what I'm trying to say. And the reason of this, இப்போ இந்தியால வந்து நடந்த recent updates, these are the updates which have been done with the biogeography of fishes. So say for example Indian banded gourami, this is the first one is Trichogaster fasciata, discovered by Bloch and Snyder in 1801. ஸோ பிளாச்சும் ஸ்னைடரும் 1801 வந்து ட்ரைகோவஸ்டர் ஃபேசிலிட்டி டிஸ்கிரைப் பண்றாங்க. ஆனா வந்து அதுக்கு அப்புறம் ட்ரைகோவஸ்டர் லாலியஸ் has been described by Hamilton in 1822. See, the Trichoaster fasciata has been described from Tamil Nadu, Arangambadi, the mouth of Kaveri. The Trichoaster lallius has been described in Hamilton from Ganges. But unknowingly, that due to confusion and the lack of awareness, the following researchers have followed that the species in India, South India, was Trichoaster lallius, and the species occurring in Ganges was Trichoaster fasciata. So we published a paper in 2022 by solving this confusion, this flaw. Actually, Trichoaster fasciata was found to be the senior synonym of Trichoaster lallius. And all the banded Gaurami in South India is Trichoaster fasciata, but not the Trichoaster lallius. In earlier or 5 years papers, there used to be a fish called Trichoaster lallia or lallius. But now it's not valid. The valid species of banded Gaurami in South India is Trichoaster fasciata. This is based on the biogeography. So Trichoaster fasciata is confined to South Indian waters. Ana Trichoaster fasciata and solli anga Gangesla solliட்டு இருந்தது பேரு Trichoaster bejius. We have also solved that confusion. So based on biogeography, the South Indian Gaurami, banded Gaurami is the species called Trichoaster fasciata, and there is no species called Trichoaster lallius, and it's a synonym. And we come to the snakeheads, Channa kelati. Channa kelati was considered to be a junior synonym of Channa gachua, but the recent molecular works from our papers and also from this paper has proved that Channa kelati is a valid species of snakehead, a dwarf snakehead under the Channa gachua species group. They have pectoral bands, which has its distribution in India and also in Sri Lanka. So India, particularly east flowing waters of South India, east flowing rivers of South India, has Channa kelati, not Channa gachua. So this paper shows that the east flowing rivers of South India has Channa kelati, but not the Channa gachua. And also, இதுக்கு முன்னாடி சில papers பார்த்தீங்கன்னா Channa orientalis report பண்ணிட்டு இருப்பாங்க. So many papers before 5 years or 10 years, you can see the species Channa orientalis. But the Channa orientalis is endemic to Sri Lanka. It doesn't have pectoral fin. அதுக்கு pectoral fin இருக்காது. But still, it was reported as from India also. We have to clarify that. It has been clarified that Channa orientalis is endemic to Sri Lanka, but not in India. The species is not in India. Rather, but we have Channa kelati in India and also in Sri Lanka, but not the original Channa gachua, which is confined to the waters of Ganges. And the Sperata avorites. So this is a group of catfish called Sperata, called the long whiskered catfish, comes under the genus Sperata. So Sperata Sperata or no species, particularly from Kaveri, they call it as Sperata or. So recent paper published from NBFGR, they have proven that Sperata avorites is a valid species which is endemic to Kaveri. But Sperata singala is endemic to Krishna. Sperata avorites is endemic to Kaveri. So this is the biogeography. So we have to understand the biogeography. Biogeography of Sperata avorites limits within Kaveri and its tributaries. So Kaveri and ella long whiskered catfishum Sperata avorites. But still now, last year papers, the reports will be there. Sperata sorry, I'm mispronouncing it. Sperata avor. But no Sperata avorites occurs in Tamil Nadu. Sperata avor confined to northern waters of north central India. What we have Tamil Nadu is Sperata avorites. The only long whiskered catfish occurs in Tamil Nadu is Sperata avorites, which is a valid one, and is endemic to Kaveri. And interestingly, butter catfish, the ompok. Tamil Nadu has 3 species of butter catfishes. One is Ompok Karingodu, one is Ompok Malabaricus, and one is Ompok bimagus. We know Ompok bimagus is common all over Tamil Nadu. But Ompok Karingodu and Ompok Malabaricus also confined to the waters of Kaveri. These 3 species exist in the waters of Kaveri. Ompok Karingodu was described by foreign researchers from the Amaravati. So it has a species, it has a black line over its lateral body. It has been named after the current code. And the Ompok Malabaricus, the Western Ghats waters of Tamil Nadu has Ompok Malabaricus. The Kaveri waters obviously has Ompok bimagus. So Tamil Nadu has 3 species of butter catfishes. Till now, no awareness has been there. Ompok Karingodu itself not recognized by many researchers. So we have 3 species of Ompok butter catfishes in Tamil Nadu. And the Ompok bimagus that occurs in Kaveri is the original Ompok bimagus, because Bloch in 1794 described Ompok bimerides from Tarangambadi. Actually, based on the topotype specimen, the Ompok bimerides occurs in Kaveri River is the original Ompok bimerides. And we come to the Murlis. The Channa Pseudomurlis, as we have seen the Channa Murlis group to Sri Lanka, in Sri Lanka. நம்ம Channa Murlis group பார்த்தோம் ஸ்ரீலங்கால வந்து அதுல ஒரு இண்டியன் ஸ்பீஷிஸ் இருக்கு அப்படின்னு சொல்லிட்டு அதே மாதிரி Channa Murlis group, Indian Channa Murlis group has had a hidden species called Channa Pseudomurlis. So India, so particularly Tamil Nadu doesn't have Channa Murlis. Channa Murlis is restricted up to Krishna. We can see that in the map in the right top. Tamil Nadu and Kerala has Channa Pseudomurlis. Particularly, this Channa Pseudomurlis is endemic to Western Ghats of Tamil Nadu and Kerala. So the giant marl, what we have is not Murlis, we have only Pseudomurlis. This is giant marl endemic to Western Ghats. And this is a species recently described from by NBFJ scientists from Kaveri River, Pungaceous icaria. So as Pungaceous silassi is endemic to Krishna, this Pungaceous icaria is also endemic to Kaveri. So this species occurs only in the upper Kaveri and lower Kaveri from Tamil Nadu and also in the Karnataka regions. So this is the knowledge required regarding the biogeography. Some species are endemic to a particular geographical region with the multiple rivers. Some species are endemic to one river. So these species are endemic. So the species so far we have seen are endemic to a particular geographical region. So they are based. They can be delimited based on their geography. Or fish எங்க இருந்து collect பண்ணாங்கன்னு தெரிஞ்சாலே we can identify the species. So that is a biogeography. With the help of biogeography, we can conclude the species that based on the place from which it has been collected. So South India எந்த பேண்ட் கவர்மெண்ட் கலெக்ட் பண்ணாலும் அது ட்ரைகோஸ்டர் ஃபேசிட்டா தான். காவேரியில எந்த இடத்துல நீங்க லாங் விஸ்க் கேட்ஃபிஷ் கலெக்ட் பண்ணாலும் அது ஸ்பெல்ட் அவரைட்ஸ் தான். அதை தவிர வேற எந்த ஸ்பீஷிஸ் இருக்க போறது இல்லை. ஸோ திஸ் இஸ் இம்பாக்ட் ஆஃப் பயோஜியோகிராஃபி. வித் நாலேஜ் ஆஃப் பயோஜியோகிராஃபி we can conclude the species based from the location it has been collected. So காவேரியில கலெக்ட் பண்றது எல்லாமே ஒரிஜினல் ஓம்பாக் பயோமெட்ரிடஸ் and based on the other two species also we have buttercup fishes and also Pseudomurlis. South India இருக்க எந்த Pseudomurlis கிடையாது. We have only Pseudomurlis in South India, particularly the waters of Western Ghats. And also the Pungaceous from Kaveri is endemic. Pungaceous icaria is endemic to Kaveri. So thank you. So any doubts can be. So now what I've told is mostly application based. So what we are doing, what we are applying in the field, I have taught you. So some may be tough to understand, but when you apply this research, when we start to work in freshwater fishes, you will understand these concepts. So this is how the taxonomy works here, freshwater, particularly freshwater taxonomy.
Thank you, Dr. Moulitharan. Am I audible?
Sir, yes, sir.
Oh, okay. So very nice and elaborative presentation on the topic which is given freshwater fishes, taxonomy of freshwater fishes. I hope that participants may have learned, because this is a newer area and where we need to spoke more. Is it? We can gain more knowledge on this area because where the information is very less. Participants, any doubts? Somebody may be in the online also, they can also get it clarified if they have doubts. Anybody from online? Okay. Since it is there is no doubts, I think we would like to thank Dr. Moulitharan for his elaborate presentation. Thank you, Mouli.
Thank you, sir.
We'll get back to you.
Thank you, sir.
Thank you, Martin. Okay, let us move to the next, next and last presentation. So the topic is molecular taxonomical tools. So I wanted to introduce my classmate who is Dr. Suresh, and who is working as assistant professor in the gate from IFPGS, okay, Department of Fish Genetics and Breeding. So I wanted to give a glimpse of about him. Dr. E. Suresh is working as an assistant professor in the Department of Fish Genetics and Breeding at the Institute of Fisheries Postgraduate Studies at DNJP. His academic foundation includes BMSc from Tanwas, and he has obtained his MMSc in Inland Aquaculture and a PhD in Fish Genetics from ICAR, CAP, Mumbai, and his dedication to that is furthering his education in evident through the accolades he earned, such as the ICAR Merit Scholarship for pursuing a PhD at CAP Mumbai during 2006 to 9, and another ICAR Merit Scholarship for pursuing MMSc at CAP Mumbai during 2003 to 4. In recognition of his academic excellence and commitment to teaching, Dr. E. Suresh received the Best Teacher Award from TNJFU, and his research endeavors have resulted in the publication of over 30 research articles in reputed journals, and he has actively contributed to research initiatives, participating as a researcher in projects sponsored by DPT and ICAR Norway Collaborative Project. He has successfully guided several MMSc and PhD students, imparting his knowledge and expertise to aspiring scholars in the field. Okay. So now I would like to invite him to present the topic which is given. Okay. So participant, please listen to him also. Okay. Thank you.
Yeah, good afternoon. Is it audible?
Yeah, it's audible.
Yeah, thank you. Thank you, Dr. Durairaja, for your very elaborate introduction about me. Yes, it is very tough time. It's a lunch time. Don't worry, all the students. I will finish on time. I have kept my presentation very simple, and it will not make you boring. First of all, I would like to thank our Vice Chancellor, Honorable Vice Chancellor, as well as organizing committee, Dr. B. Ahilan sir, Dean sir, and Dr. N. Jayakumar sir, Professor and Head of the Department, Dr. Durairaja, Dr. Sudhan, for giving me this opportunity. It's also my pleasure to share the platform with one of the greatest teacher in as far as our institute or university is concerned, Dr. V.K. Venkataramani. Just before half an hour back, he delivered a great lecture. Listening to him, it is always my pleasure. Thank you very much, sir. Now let me share the slide with you guys. Hope it is visible. Anyone, please? Is it visible, Dr. Durairaja?
Yes, it is visible.
Yeah, thank you. Yeah, thank you once again. The topic that I have been given is application of molecular tools in Ichthyotaxonomy. So the first slide is nothing much about that. Why the fish identification is very important in fisheries point of view. So these are the very important point. The accurate species identification is very useful for evaluating biodiversity, regulating fisheries, identifying adulteration in seafood, as well as authenticating seafood. Overall, the fish identification is very much important for managing the fish genetic resources of India. But out of all these problems, these two, that is identifying adulteration as well as authenticating seafood, is the topmost problem in the world, right? For this, how the molecular tools are used, we will see. So generally, the identification method, particularly in fish or Ichthyotaxonomy, this can be broadly classified into three. That is traditional based method, protein based method, DNA based method. This has been exhaustively covered by previous speakers, Dr. V.K. Venkataramani, as well as Dr. Moulitharan. Just have given very few things, and the traditional usual morphometric characters, autolyth based, scale based, or in many things are there. And protein based markers that comes under, there are markers, that is isoelectric focusing, capillary electrophoresis, HPLC, ELISA, RFLP, RAPD, AFLP, microarray, LAMP, DNA barcoding, and eDNA metabarcoding. Out of all these DNA-based markers, the most common the two markers used for fish species identifications are DNA barcoding method, or otherwise known as mitochondrial based DNA marker, and eDNA meta barcoding. These two things only we are going to discuss in this lecture. Why DNA based methods are important? It has been proven that the DNA based method is most powerful one, as well as because due to its flexibility as well as very speediness, that is rapidity, and as well as cost effectiveness. Moreover, the DNA is also thermostable. It can tolerate some of the wide range of temperature and relatively stable. So not only that, it can also identify any processed products at a higher resolution than protein or morphometric based. These are some of the reasons why we should go for DNA based method. Method is better than protein based or traditional based methods. Now let us discuss about DNA barcoding. So this is we will see in detail. So this is the man. Because of this man, his name is Dr. Paul D. N. Hebert. He's also known as father of DNA barcoding in the world. This man is basically from Canada. He has established Center for Genomic Research. His expertise in all molecular work, particularly evolutionary relationship among the organisms. He is the one who established this DNA barcoding concept. Since then, an n number of papers have been published. A lot of works are going on. So let's go now. This is the general workflow of DNA barcoding. So you can see just a very simple manner. So you need to collect the sample first. Then you need to extract some of the tissue from the sample. It should be stored in 95% ethanol. Then from the stored tissue, we have to isolate the DNA. Then from the DNA, we should go for PCR reaction. After PCR amplification, the PCR product should be sequenced using Sanger sequencing, and then data should be analyzed for the species identification. This is the general workflow. Now let us see one by one. The sample, while collecting the sample, it should not be any contamination with other tissue or any other species. Then if it is so, then it will lead to the wrong identification. Then once collecting the sample, that should be or must be identified with the standard reference book. Morphometrically, that species should be identified. Then the approximately 50 milligram muscle should be collected. It can be either muscle tissue or fin clips. Then it can be preserved in 95% ethanol. It can be stored in -20 degrees Celsius. Then not only that, the samples that are collected from wild or whatever, wherever river or marine areas, that should be stored in 10% formalin as a voucher specimen. That should not be discarded. It should be stored in formalin. These are very important steps. Then the next step is DNA isolation. There are many standard protocols are available. Mostly the protocols that are used, phenol chloroform method as well as salting out method. So once the isolation in DNA, the quality and quantity also should be checked. Then only we can get the PCR product that can be checked through UV spectrophotometer as well as nanotrap. Then once you, after isolating the DNA, we should go for PCR amplification. There are many primers available for PCR amplification. Universal primers are available. The one that is shown here is the universal primer for particular region, that is CO1 gene region, that was discovered by Ward et al. In 2015. The length is approximately 650 base pair length. Then not only that, while doing PCR amplification, the steps that are PCR condition, that is 3 steps, denaturation, annealing, extension, these things should be standardized for getting, otherwise we will not get any PCR product. So here you look at this this table. These are the, for DNA barcode, number of primers are available. See, fish F1, fish R1, fish F2, fish R2, like that. So many universal primers are available. These are for which region it targets. CO1, 16S. This is the most commonly used. These are shown here. The target organism also. For example, so you look at the crustacean. Crustacean, the primer is known as max F, max R. So the primer sometimes it varies from fish species or molluscan region or crustacean. Standard have to choose the primer regions. So this is also thermal profiling also they have given. This has taken from this author. So once the PCR successful amplification, this should be checked in the gel documentation system. That should be sent for DNA sequencing. We can receive the sequencing. After sequencing, we have to check the sequencing quality like this by using sequence scanner. There's a software called. From that we can see there's a score called Fred. Should be more than 20. If it is more than 20, all the sequence are good. So then we have to check. Once the checking, after checking that one, that should be similarly checking. We should check in the online tool that is called a BLAST. There it will show the how much maximum where it goes and matches with. For example, here in this the sequence, it goes to 100% or more than 99% and 100%. If it comes, we have to select that the sequence is correct. We can select the species. This is the blast result will show. These are the information, species name as well as maximum score, total score, e-value, all these things, including accession number and total length of the sequence also will be shown. Once after the blast, you can use the same sequences using bioedit software and mega software. You can develop among the organism on the fishes, selected fishes. This will give the phylogenetic tree. Just before the previous speaker shown, the phylogenetic tree using these sequences can be constructed using the software called Mega. That is now latest version, Mega 11 point something. This is the Mega software, molecular evolutionary genetic analysis, that is written as Mega. The software looks like this only. So now the DNA barcoding, if you look at the DNA barcoding papers, if you last 10 years, if you select, before that, you should understand the mitochondrial DNA structure of the gene, the mitochondrial DNA structure of the fish or animal. It's all more or less similar only. For example, in fish, there are 37 genes are available in this mitochondrial DNA. But out of all 37 genes, mostly DNA barcoding, they have used 12S RNA, 16S RNA, D loop region, NDA region. You see here, cytochrome B, D loop, 12S. This is, but most commonly used region for DNA barcoding either cytochrome oxidase 1 or cytochrome B. Cytochrome B. This, you look at this cytochrome B as well as cytochrome oxidase 1. Only with this small region, see the length of the entire mitochondrial DNA structure is, this is the base pair. 651 base pair is the for fish. Okay, but this region covers only 650 base pair. Only within this region, between species or among the species, there is a variation. Based on that, the DNA barcoding has more than 15 years. Paul Hebert worked vigorously in his lab. After that, only he came to the conclusion this region can be the particular choice for the marker choice for DNA barcoding. Only with this small region, we don't need to sequence all the regions and compare whether the two are different or not. It's not necessary. Only this small portion, that is 650 region, 655 base pair alone, can be used for distinguish the species. That is what the Paul Hebert established. We are doing DNA barcoding for species identification. If you look at this table also, the Bhattacharya and the that's little old from the 2015 paper I have taken. See the number of species available in the region wise they have given Arctic, Antarctica, Africa, including India. These are the not fish species, the total number of species here. Fish species specifically, I did not get the data. But how many species available, how many species have been barcoded here it is given. So you look at this in the percentage wise it is given. Only Arctic regions have been covered more than 75%. Because the number of species that is available only 240, so they have barcoded 178. In India also significantly not significant. See, look at the India percentage. Out of 11,000, we have completed only 1,900. It is 2015. Maybe it is little more recent. So here region-wise they have given barcode progress. Therefore, still so many species in the inaugural speech itself, Dr. Venkataramani sir as well as Dr. Ahilan sir and Dr. Jayakumar sir also said how many species are available in the world. New species every day it is discovered. The taxonomists are not available. Therefore, so many years it will take many decades to identify all the species. Even though after developing DNA barcoding technique also the progress is very slow, but we cannot reach the speed level. It will take another 2 to 3 decades to complete all identification. So in world over, for exclusively, there's a system called barcode of life to barcode entire species in the world, including microorganisms, butterfly, many organisms. They have established barcode of life platform. Under the platform, they have established the fish barcode of life initiative also. Under this initiative, under this initiative, whatever the barcode is done, sequence are deposited in this platform. This serves as a reference database for fish species. So these are the 4 common fish barcode of life initiative that is called fish bowl, French Polynesia fish barcoding base, aquagen, and GenBank. Among these, mostly scientists or students utilize the GenBank platform as well as Fish Barcode Life Initiative. They use for as a reference database. So another one important reference database many people might not be knowing. This is established in India under NBFGR, National Bureau of Genetic Resources in ICR Institute is located in Lucknow. They have the Aquatic Genetic Resources Information System of India, exclusively exclusive to India. See, for example, shellfish, fish photo gallery. You can add new species, including GIS map, fish nutrition profile. If you go and type the name, you can find out the species that are available in India. You can get all the details. So therefore, you can go to NBFGR, you can click this icon, accuracy, then you can get all the details. For example, this data latest in today's morning also, more than 3,200 species are available under the genus of 1000, genus family 256, under 57 order, type specimen, the photograph, more than 7,000 are available. This is the very important database in India is concerned. That is NBFGR is serving as a nodal agency for keeping all data as a reference data. So the main topic today is only species identification only. The DNA barcode is very much utilized for mainly species identification. In addition to fish species identification, the DNA barcoding can be used cryptic species as well as can be used to identify fish egg, larvae, including fillets, including fingerlings, as well as pathogens. And the invasive species is one of the very much problem in fisheries management concern that can be also identified, including endangered species, forensic case, mislabeled food product. These are so many applications. The same what I expressed now, the protocol, you can use the same protocol, but you can identify all the species. So some of the papers that was published for species identification, this is one of the very important paper, DNA barcoding in Australia for species. Very earlier, this paper was published. Number of species, more than 61 species they have published, including Western Himalayan cold water fishes also were identified using DNA barcoding. There's another important paper that is DNA barcoding of Indian marine fishes. Here, more than 100 species were identified by DNA barcoding technique. This was established by, this is as far as India is concerned, this is the person, Dr. W.S. Lakra. He is the one who started DNA barcoding of fishes in India. He is retired. He was the former director of NBFGR as well as CIP. So this another one is, for example, the another application that is called the last speaker, Dr. Moulitharan was speaking. Cryptic diversity also can be identified through DNA barcoding technique. For example, in this paper, DNA barcoding reveals cryptic diversity. They have collected 160-30 specimens. Out of the 20 species, 20 considered are valid local species, 2 identified as cryptic species. Using DNA barcoding, they could find out that the another 2 species is this. So out of 22 species, 2 cryptic species they have identified. There another one also here in this article, DNA barcode, they identified just eggs, fish eggs for 25 species belongs to 11 families and 2 orders. The identification was very successful. More than 99.78% the success rate was. At most 100%. There was no misidentification. So the DNA barcode to serve to identify fish eggs also. This is, I think Dr. Moulitharan was speaking about the Pancasia silasi. That is the one, this is species. I am happy to share that I am also part of this important identification. My name is here for your just reference. This is the fish Pancasius. These are the fresh one, preserved one, and these are the same species only. I will tell you how this species were identified. You look at this upper proof. If you open the mouth of the species Pancasius, you can distinguishly see that this was the only work I contributed for this species. My name was here. See, this upper roof of the this species drawn here. You look at this ventral proof, upper plate, ventral plate, upper plate, ventral plate. Compared to these two, look at this ventral plate. Here there is no joint. Here there is no joint. But here there is only one ventral plate is there. This was the only fish I took, I drew, I'm happy to that. Just I could identify this. So how we can identify fish species? These are the rules, including this roof pattern also serve as to identify cryptic species. In addition to the DNA barcoding also supplemented just to identify the cryptic species. So this was identified, then it was published in the hydrobiology in the year 2017. I am also part of this. So another one, this is another very important paper, that is DNA barcoding reveals species of composition and sharks and rays. There was a case in Kerala. So shark fishing was banned, but however, they came to know that fishermen were catching the shark and cutting the fin and mixing with other fillets. They were just transporting and selling illegally. But the case came to NBFGR Cochin. They collected the samples. They identified, then they did the DNA barcoding. They could identify that sharks were endangered one. The fishermen were doing illegally all the trade. Then court finally just sentenced them with a fine along with the jail. So the publication is this one, the great contribution from the NBFGR on shark trade also. That means fillet and illegal trade also can be found out by DNA barcoding. This is another one paper, mislabeling in Indian seafood, an investigation using DNA barcoding. This is one of the alumni, Dr. Nagalakshmi. She published from the CIP. She collected more than 100 seafood samples, including fresh, frozen, ready to cook, ready to eat, and canned. So many types of all varieties she collected from all over India, particularly in metropolitan cities. Found out that 22% of the seafood mislabeling. That means it may be actually it may be originally it is written pomfret, but maybe fish may be a pangasius. Like that, 22% of the seafood were mislabeled in Indian domestic market, not in the export markets. So this is how the DNA barcoding is useful in finding the mislabeling in Indian seafood also. So is there any limitation? Always it's not advantage. There is a limitation or criticism of DNA barcoding also. So this DNA barcoding can be used to only the recently diverged species only. So species in is very much important. If it is very recently established or happens, it is very difficult to identify where barcoding cannot identify. Then not only that, even hybridization also happens among the fishes. If the hybridized fish also very difficult to identify DNA barcoding, this can be identified along with other DNA molecular markers and along with the protein marker also. So incorrect identification of species also sometimes lead to wrong or incorrect sequence. This is also limitations of DNA barcoding. So some of the DNA barcoding has been done in our TNJB also. This work was done in the Pulicat Lake in Chennai. There around 29 species were collected and identified DNA barcoding successfully. The success rate was 100%. Then some more work were done on Nemipterae family also in TNJFU. Next moves to the another normally or commonly used molecular tool for fish species identification, that is eDNA metabarcoding. So what is eDNA? It's called environmental DNA. What is environmental DNA? It's a definition for environmental DNA is any genetic material obtained directly from environmental samples that includes soil, sediments, and water without any obvious signs of biological source material. That means DNA can come to water or soil through how it can come from fish skin, mucus, saliva, sperm, secretion, egg, feces, or urine, blood, through which in DNA are mixed in water, soil. So the soil or sediment, water can be collected from the DNA, can be extracted from that, we can identify fish species. That is how the environmental DNA or eDNA metabarcoding came to the light. The eDNA metabarcoding for macrobial organism, not it. It used to, the technique eDNA was first utilized for microorganism only. But macrobial organism, it was first used in 2008. Since then, it has been utilized in fish species identification also. This is the general protocol eDNA methodology. We have to collect the sample, either water sample, sediment, or soil. Then from that we have to extract the DNA. DNA can be amplified using universal primers. Then this is the very important phase. After amplification, amplified products should be sequenced using next generation sequencing, not Sanger sequencing, because we are going to identify multi-species here. Then from that meta-barcoding, we based on the reference database, we can find out the species list. We can find out more than one species from this DNA meta-barcoding. So sample collection, this is called eDNA sampler. This sampler costs around 10 lakhs, very recent quotation. We water samples, or you can collect water through gram sampling or composite sampling also. Generally the volume, how much volume we should collect, 15 ml to more than 5 liter, or even up to 68 liter we have to collect water. From that we can filter the DNA. So volume may be related to the DNA, eDNA. The fish eDNA, how much is there, we don't know. So the volume depends on the eDNA level available in the water also. Generally, the standard sampling volume for streams, rivers, if we do eDNA sampling in the streams or rivers or lagoons, the water volume is 1 to 2 liter is sufficient. So this is the eDNA sampler working procedure shown how to collect the water. This is how the eDNA sampler looks like. There is an on and off button. From this portion, the pole end of the portion from the water is suction is done. Here you can control the water level, the speed, flow, pressure, all they can be checked. Along with the water sampler, eDNA sampler, they give filtration, scissor also. So the filter is fixed end of the pole, end of the portion. Then the water section is controlled by a remote control. So once the water section has been done, then we have to remove that filter. The filter that contains that filter as such is preserved in ethanol, or then it is transported to the laboratory for further analysis. So the eDNA capture only we have shown. Filtration is the most commonly used method for capture eDNA. We have to collect the sample. See here in this, there is a filter. It is that the water is sucked from the vial. It goes like this. While going water, the filter is filtration is done. It captures the eDNA. The membrane filters are mostly used. The pore size of the filtration is very important. Generally, pore size is 0.22 micron meter to 3 micron meter. So not only that, the filtration medium also very important. There are a lot of standardized glass fiber filters, cellulose nitrate filters, polyethylene sulfonous filters are also used commonly, but mostly glass fiber filters gives good result the survey has done. After that, eDNA can be extracted from the filtration. There are many kits are available through which only we have, so far the literature say commercial kits can alone be used to extract the eDNA from the water samples or soil sample. The most commonly used kits are DNEC blood and tissue kit, and as well as we can use CTAB method as well as PCA also. So this here, you look at this, how many extraction methods are there? Salt extraction, rapid water DNA isolation. These all commercially available kits. So among these, you see this one, DNEC blood and tissue DNEC tissue kit. So the power isolation kit they have used. So mostly DNA extraction, the kit that is used is DNEC blood and tissue kit. So once after the extraction of DNA, amplification should be then PCR amplification. After using the universally available primer, for that also so many primers are available, M13, M14 like that. We can go for any kind of PCR, conventional PCR, qPCR, nested PCR, digital PCR. Then real-time PCR also we can do. Then we have to sequencing using next generation sequencing, any platform we can use. Based on that, then we can use for the similarity index to identify all the species. So look at the primer names. These all the universal primers, so many are available, more than 25, 30 primers are available for which system they have established, freshwater, cartilage, even group wise also they have established, as well as group wise also marine fishes, freshwater fishes also it is there. So these are the primers according to our union we can use and standards. So what is the application of eDNA method barcoding? The first application, as far as this seminar is with the World Taxonomy Appreciation Day, the species, a single species identification also we can use. Multi-species also can be identified. It can also detect rare species as well as like what DNA barcoding does, like that eDNA method barcoding also does. but it is in the group. There we can species only, but in case of eDNA metabarcoding, without taking tissue, you can, based on environment samples, you can identify n number of species. Some of the example for you here. So in this, one of the studies has been done in the backwater lake. Okay, so this is the structure where they have collected different places, all water samples. They have compared a traditional catching method as well as eDNA metabarcoding. So the number of species that have been detected by traditional catching method as well as eDNA metabarcoding, only 1 or 2 is missing in this, not detected in eDNA metabarcoding. See, look, here in this method, you have to collect the fish samples, you have to collect the tissue, you have to identify. But in this case, you don't need to see the fish at all. You have to collect only water or sediment or soil, that's all. From this, it could identify almost more than 98% of the what traditional method catching has been identified. Also, eDNA has been identified. So, what researchers say, eDNA metabarcoding very well we can use along with catching methods, traditional catching methods for fish species identification. So, another study is shown the comparing capture-based method as well as eDNA metabarcoding. You look at this in this, out of 18 species total to this to 18 species, 12 fish species were identified by eDNA metabarcoding. Okay, all together, so so it is 18 species. The 2 species can be collected from the common, but out of 18 species, only 6 species could be identified by capture based method. These 12 species, among the 12 species, you see non-native species recorded before, species not recorded before and detected in several sites, species not recorded before and only detected 1 or 2 sites. These are the only native species. So the species that were not recorded before also could be identified by eDNA metabarcoding. This studies has proven that in a very recent eDNA metabarcoding can be identified. See, those people who were recording earlier, they could not see the fish, they could not record. But only based on eDNA, they could record non-native species are available like that, they have listed out. Another detection, you see this one, eDNA based on morphometric as well as eDNA species wise in the studies they have done in the deep sea fishes. This particularly this study was in the deep sea. See, eDNA could have species, but morphometric could identify only 4 species, 2 are common. Like that in genus wise also it is 16 genus could identify, it is only 5. Family wise it is eDNA 10, morphometric only 6. It shows that this paper, the last 2 papers shows that eDNA metabarcoding is better than traditional method for identifying fish species. So another these studies, you see this green shows only by survey sighting, only 2, 4, 6 species, 7 species are there. This blue line indicated, including whatever the species, the number of species are available, could be identified by 12S eDNA metabarcoding. This one is, the red one is CO1 eDNA metabarcoding. This is what available in the black one is in the museum. So compared to all these things, definitely eDNA metabarcoding identify more species than traditional method. This paper also very recent, it's a 2021 published paper shows this indicates that eDNA metabarcoding can be very well utilized for fish species identification. So, lecture, madam. This is another important point we have to consider, that factor influencing the DNA, eDNA. It is always the we have to note it down that we cannot our expected result always. So many factors that affect eDNA metabarcoding flow. So see, look at this. How many factors? This is the eDNA. Fish releases DNA into the water. It is transported. It is degraded. It is sedimentation. It's settled. The factors that influence. See, eDNA can persist in water for days to months. But what affects this eDNA quantity or quality or availability? Abiological factor or abiotic factors, including hydrological condition, water temperature, UV radiation, pH, salinity, substrate, turbidity, and water chemistry. Biotic factors that can influence the eDNA is, if the more organisms are available, the chances are that more organisms, more eDNA we can find, because more organisms are there, it will shed more, it will release more eDNA, including life stage behavior. The larval may not release that much eDNA, but bigger animal can release more. So body size, metabolism, the stress organism or under seasonal migration, these all factors that influence the eDNA. So what challenges in eDNA metabarcoding we can see, it is always that while collecting the samples, the contamination occurs. So the contamination lead to the wrong identification of a species. This is one of the important challenges in eDNA metabarcoding. Not only that, while collecting eDNA, there is a humic substances are available that inhibit the quality of the DNA, to degrade the DNA, so that also does not give the results. Not only while is another important factor. Sometimes we get erroneous DNA sequences that leads to the wrong identification. So as I said, that some of the reference data of DNA databases, DNA barcoding databases are available, but here now very recently established some of the only few reference databases available. Therefore, identification is somewhat difficult in this. So sometimes interpretation of the results also goes wrong in that we identify wrong species. That also one of the challenges in the eDNA metabarcoding. I think in the summary, I would say that so molecular based techniques are the most powerful technique in taxonomy. However, without the support or without the complement of traditional taxonomy, the identification of fish species will not be sufficient. Therefore, integration of traditional taxonomy with molecular based taxonomy is always good for fisheries resource management. I think that's all from my side. Thank you very much. Any clarification if you need, just we can.
Thank you, sir. Participants over online, if any doubts are there, you can clarify.
Yeah, please.
Hello. Hello, sir. Hello.
Yes.
Hello, sir. My question is regarding the environmental DNA, sir. Suppose that take the samples of our environmental DNA. The main challenge is, sir, how to segregate, sir, that sample, sir.
So from the tissue paper, how to segregate the DNA? You're asking that one only, no?
Yes, sir. Yes, sir.
Yeah,
How
that is
to segregate?
only using the slide scraper. We are doing like mucus select from mucus. We are collecting from fish. Like the tissue paper, we can segregate. We can transfer to tube, a pond of tube.
Thank you, sir.
Okay.
Thank you, sir. And now we have came to the conclusion part. So a national webinar on Ichthyotaxonomy, Foundation of Fisheries Science, was organized in commemoration of the World Taxonomist Appreciation Day by the Department of Fisheries Biology and Resource Management at Fisheries College and Research Institute, Thoothukudi. So total we have around more than 100 registrations from students, researchers, different parts of our nation participate in this program. So of which 54 student representations were attended in offline, the hybrid work. So Dr. N. Jayakumar, sir, Professor and Head, welcome the gathering. Dean Dr. Ahilan, sir, delivered the inaugural address and emphasized on the importance of taxonomy and the need for following the taxonomist. And Dr. D.K. Venkataramani, sir, former Dean, Adjunct Professor, served as the first speaker and explained about the marine fish diversity, classification, identification of marine species, and the criteria to be followed while electing the new species or revising classification of species. And the second speaker, Dr. N. Moulitharan, Assistant Professor. He highlighted the need for exploring the inland fish diversity, classification and identification of endemic and cryptic fish species. Of course, the need for revising the classification of certain species for resolving the ambiguities in their classification. The last speaker, Dr. Suresh, Assistant Professor and Head in Charge, he focused on the various molecular methods available for the fish species identification and the fish logs. So I thank everyone. I thank all the speakers who served to speak up for today's function and the participants from various parts of our country. With these words, I conclude the webinar, and the participant certificate will be distributed to the concerned mail within 3 working days. Thank you.